picture can also, okay, recording in progress. Thank you. This rather bleak picture can also be turned around because, as we know, agriculture is also a potential contributor to the solution of many of these crises. Agriculture can contribute to climate change mitigation in that it increases the humus content in the soil, and that it stores carbon in the soil. It can also contribute to biodiversity conservation and can improve and foster our cultural heritage. It can protect water resources and protect the soil resources. But for doing that, for doing all this, we first need to, we need to see that some of these aims, at least in parts, are sometimes conflicting. So the question is, how can we reconcile these aims? And I think what is currently evident is that agriculture in its current form is unable to solve all these questions or contribute simultaneously to the solution of all these different crises. So what we need really is a global and very thorough transformation of agricultural systems. And one aspect of that transformation could be provided by making agriculture more biodiverse, more increasing diversity in these agricultural systems. And that's what I'm going to talk about. And throughout my talk, I'm going to try and refer to individual solutions that we make to individual problems here highlighted in this picture, where we can really see that we provide solutions for the biodiversity crisis and at the same time can also increase productivity and at the same time really provide protection of soil and water resources. So I'm going to structure my talk according to these 5 topics. I'm basically looking at increasing plant diversity at increasing levels of complexity. We will begin with minor crops, basically adding crops to the cropping systems that are only rarely grown, but that can provide very good services. We are going then to expand diversity by mixing cultivars, mixing crops at the genetic level, and then going on further to increasing the complexity by mixing different species. And this is already the point where we can see very complex interactions going on between the different partners that will further increase with agroforestry, and then that will culminate in the approach of permaculture design. So the first approach here that I'd like to mention is the adding of, I've called them new crops, but they are actually not new. They are very old crops. They are just not very often grown. They are medicinal plants, for example, or plants, herbs, for example, oil crops. All of these that are depicted here in this slide have in common that they are flowering and they attract insects, flower visiting insects. So the idea here is to use the full spectrum of crop species rather than only a few species that we are currently concentrating on at the global level, so that we can provide higher dietary diversity, first of all, but also, of course, higher diversity to all the other organisms around us, so to the insects that are visiting these crops. But perhaps importantly, at the farm level, there's also, by introducing new crops, there's also a reduced risk of complete failure. So if a farm is affected by something like extreme weather events, then by having more crop species on the farm, we are insured against the risk of failure to a higher degree. So that already is a kind of adaptation to climate change to increase the diversity of these crops at the farm level. And of course, it's also possible to replace some of the fossil fuels by plants. I believe that Thorsten Kraska will later on talk about this, but the link to biodiversity here is very important. So because we are able to provide resources not only for ourselves, but also for the insects that are visiting these flowers. And this beautiful picture of a poppy field at our organic experimental farm shows that it's also beautiful to look at, but some research shows here, this is actually not for poppy, this is for caraway. This is a pollination network, or to be more exact, a flower visitation network. And I've highlighted the caraway here. So you can see on the right-hand side, all the different insect species, actually only half of the different insect species that have been recorded visiting this one plant species, this caraway, that was introduced into the cropping system. And on the other hand, on the other side here, on the left-hand side, you can see all the other different plant species, the wild plant species that are concurrently visited by all the flower visitors. So there's a really complex network here, and we are providing resources for the whole ecology of this flower visitation network. Now, I come to the next level, and this is about cultivar mixtures, mixing different crops at the genetic level. So what is normally done is that we have large fields that consist, especially in cereals, that consist only of one genotype. So basically that means all the plants are genetically identical within that field. I've shown here two varieties from a variety trial in the north of Germany, where wheat is grown with very high intensity, and there's actually no diversity within that field. But normally, if you think of the traits of the properties of these cultivars, then it's actually not. So one important point is that the maturity of different plants in the field should be the same, so that they can be managed and harvested at the same time. So it wouldn't be a good idea to mix different crop varieties that are maturing at different times. But once that is provided, there's really no good reason why you should only grow one genotype in the field. And I've highlighted here two examples of growing different genotypes, different cultivars of the same species. In the left-hand side, this is rice, and here we've got a species of wheat from the US. And here in the rice example, you've got two different varieties. One is glutinous, high quality, very precious cultivar, but it's also susceptible to certain fungus. And the other one is a more standard variety that is, however, less susceptible, that is more resistant. And that means that if you bring these two varieties together, then you can protect the more precious variety, the more a higher price variety that is glutinous, that has a higher quality. You can protect it against the fungus, and you can then also save fungicides and then reduce environmental pollution. And very much the same is the case in the wheat, where certain rust funguses can infect and reduce the yield of these crops. But by mixing the different cultivars, we are protecting the more susceptible one, because the fungus cannot travel so fast between the susceptible individual plants. So what we also then can see is that by mixing these cultivars, we can increase, only slightly, but we can increase the yield. Here we see the relative yield of the mixture in relation to the monoculture. So at a point of 1, we see that there's no difference here between monoculture and mixture. And on the right-hand side, the mixture is better than the monocultures. And these points here represent the data where we can see that with mixing 2 different varieties, we get 2%, so 1.02, 2% more yield. Also in the same region with 3 cultivars, but that can actually be increased furthermore by including more cultivars, by mixing 4 or more cultivars. So 5% more yield there. That is not a lot, but the main point here, by mixing different genotypes at the field level, is that we can very reliably reduce diseases through various mechanisms. One is that the fungus cannot spread so fast in the field. There are also other mechanisms that we can perhaps discuss later on. But there's also an important, further important point, which is that the higher genetic diversity increases the yield stability of the crop, which is especially important in adaptation to changing and fluctuating weather conditions in climate change. So, as I said before, the mixing effect tends to increase with the number of mixed varieties. And the last point that I'd like to highlight there, mixing different genotypes at the field level, is a slightly different aspect. Here you can see not variety mixtures, but crop populations, where actually here on the right-hand side, every plant individual is genetically different from its neighbor. So we've got a huge genetic diversity. And the point there is that we take the seed from that ripening crop and plant it in the next generation. And the idea is that by doing that several generations, year after year, we can generate a crop where through natural selection, those genotypes that are most adapted to that cropping system and to that site are going to increase in their frequency in that population. So that means over time, the crop is really adapting to the prevailing environment, and combined with the ability of higher stability, of buffering against environmental fluctuations, these two points are really important properties that protect us against the vagaries of climate change. Only, let's say, to a certain degree, this is certainly not enough in terms of adaptation, but they can contribute some insurance and some adaptation potential here to the cropping system. So we go one level up and mix different species. And what you can see here on this field experiment is, on the one hand, here on the right-hand side, a mixture of wheat and bean, fava bean and spring wheat. And on this other plot here, next to it is pure spring wheat. And somewhere in that field, there will also be the pure fava bean. So by bringing the two different crops together, we have an explosion of different interactions between these two partners. And some of the mechanisms that may occur are listed here. So one partner, for example, the legume in this case, may provide nutrients, in this case, nitrogen, because the legume is able to fix nitrogen, and it may transfer this nitrogen to the other partner, to the cereal, so that the overall resource use is much more efficient in terms of nitrogen use. Yeah? So the mixture is more efficient in using nitrogen than the two monocultures if they are grown separately. Another example, which is perhaps less known, is that one partner may improve the accessibility to nutrients for the other. So in this case, in the legume cereal partnership, this works the other way around for iron, because iron is more accessible to the cereal and can actually provide iron or make it more accessible, more increase the uptake of the iron by the legume. That has been shown in many different studies. A further point that I've already mentioned with respect to the cultivar mixtures was that the disease and pests can be reduced, because in this case, the density of the susceptible plant is reduced so that the pathogens can't spread so fast in the crop. And also what we've already seen in the cultivar mixtures is a kind of compensation, because if one partner fails, then the other one just takes its place. So if you imagine that for some unfortunate reason, the faba bean in this plot here completely fails because of a let's say a soil-borne disease, then at least there's the wheat there to take up the space in between, and it will compensate the failure to some degree in the mixture. So there's an insurance against failure. And also this last point, the point of complementation, the gaps left by one component are filled by the other partner, which is then leading to better resource use overall. So what we see is, if we take all these different mechanisms together, is that we have a higher productivity in the mixtures than in the monoculture. The yield is also more stable. They tend to be less infested by diseases and pests. Of course, this is not always the case, but on average, it has been shown in many studies that there's a benefit, and also they need less fungicides, less insecticides than the monocultures. In some cases, and this effect is actually slightly less pronounced than the other ones, they can also suppress weeds better. That maybe depending on the density of the crops. But as I already said, the resource use is more efficient because they basically of this overlapping niches of these two different crops. They may support more pollinators. I show you a picture of that, or some evidence of that in the next slide. And other research shows also that they can also provide larger food range for other insects. And if you take all this together, basically you've got multiple functions. So diversity actually provides more functions or higher performance in multiple functions, so that overall there's a higher multifunctionality. And just as one example of evidence for the higher productivity, I'd like to show you this picture. The LER is an index of that compares the productivity of the mixtures and against the monocultures. So the line here at 1 signifies that there's no advantage of the mixture in comparison to the monoculture, whereas everything above that line means that the productivity of the mixtures is higher than, in this case, the yield. The yield is higher of the mixtures in comparison to the monocultures. And you can see here that in this data set that we analyzed with our PhD student Demir, with many crops, there's actually higher productivity. And wheat, there's an overlap of this this line, but these are only a few studies there. So you can see that really here for the cereals and also for the legume partners, overall, whatever you look at, of course there's some variation, but overall and on average, there's very reliable higher productivity in the mixtures. So here's another example of the benefits of such species mixtures in this picture that I've shown you before with the wheat and the faba bean. Here we look at the infestation of the faba bean by these aphids, the black bean aphid, which can sometimes be very damaging to the crop. And you can see here in the organic and the conventional system at high and low density, in all cases, the monoculture had a much higher percentage of aphid-infested plants than the mixture. We know also something about the underlying mechanisms for that. That has probably to do with the host finding of the aphid in this case. But this is really reliable, and we can then, if we then further rely on natural enemies to further reduce the populations of these aphids, then we actually can do completely without insecticides. And this was happening in the conventional field, and the technicians there was really surprised to see that all the insecticides were completely needless in this case. Okay, and there's another example here for benefiting, in this case, benefiting the insects. Yeah, so we combined different legume species here in an all-species mix containing 10 different legume species, whereas the control mixture contained only 2 different species. And from many different pollinator groups, we could find, or for some at least, there were some significant increases in the flower visitation rates for the more species diverse mixture than for the control lay. But perhaps importantly, these same mixtures were also different in their productivity, in that the higher diversity also provided more biomass, more forage for the, or more fodder for the animals. So here we can actually break up this conflict between biodiversity protection and productivity. We can simultaneously increase both. Yeah. So next level up is agroforestry. So we still mix different species, but what we are doing here as well is also very much increase the structural diversity of the whole field. Here's a young agroforestry system in the south of England. You've already seen the farmer in the previous slide, and he's called Tolly, and Tolly says he's farming biodiversity. Yeah? So, and that's what he's doing here. Also for protecting the soil and for enriching carbon, but also, of course, because he's further increasing the number of crops and thereby ensuring against failure. And one important point that was highlighted by a farmer that introduced agroforestry in Bavaria onto his organic farm, one important point here was that he always made. This is Sepp Braun from the south of Bavaria. He said, basically, if we look at what we are growing normally, which is to a large degree cereals, then at the point where the number of hours of sun hours is greatest in our region, then there's actually no photosynthesis going on. Yeah, so we are actually not making use of that light level. Yeah, there at that point where also the sun, the angle of the sun is highest, so where we can actually the irradiation is highest. Yeah, the crops are already at maturity. Of course, this is going to be used in a way that promotes the maturity of the cereals. It's not a bad thing, but we could make more use of that resource. And one idea is to plant trees to harvest that sunshine. Especially because in that case, you can imagine that the trees in an agroforestry system, as you have seen in the previous picture, if the trees are a bit larger, then they will shade the annual crops. But for the cereals, actually, that doesn't really so much matter in that time window where the sun is highest, because the shade is then, because the photosynthesis is then actually more or less has ceased. Okay, and you can see that here in a simulation study from France, where you've got pure trees, that is walnut trees on the one hand, and a pure cereal crop over different years. And you can see here how much light is going to be used by that cereal. In total, the black bars signify, they mean the sunlight that is actually not being used. Yeah, so you can see here 25% of the light is going to be used by the cereals. And on the other hand, the pure trees, of course, over time they grow, and they the green area here shows how much light is going to be used over time. And as the trees grow, more light is being used. But the point here is, if you mix the trees and the cereals together in an agroforestry system, then you can actually reduce the amount of unused light very much. You also see that the trees are going to shade the cereals to some degree. There's some reduction in the light level that can be used by the cereal. But overall, and that's the main point, the resource use is much higher, in this case 39% higher of the agroforestry system in comparison to the pure crops. So there are many different ways of designing agroforestry systems. I've highlighted here two of them. There's a silvo arable system here in France, where the trees are grown with cereals, but you can also combine, of course, these trees with pastures and with livestock systems. There are many different ways of arranging the trees on the area. Most of them that I've seen are arranged in these alleys. That makes it a bit easier for the machinery to go in between the rows. But there's really no stopping the creativity for designing these agroforestry systems. And perhaps one more important point is here the protection of the soil. You can see here a slopy ground where the trees have been planted at regular intervals to stop soil erosion at this slopy field, so that you can actually provide the resource protection that you need for continued cropping of that area. And if you increase the normally you find these agroforestry systems with a very high complexity in the tropics and subtropics, and you could summarize the effects of these agroforestry systems in that you've got a higher productivity. I think I've shown that some of the reasons for that in the previous slides. You also find that there's a tendency for increased biodiversity because you provide more habitat. You can also increase the carbon storage, and you've got, as I've shown in the previous slide, less soil erosion. So you really have a multifunctional high performer here with the agroforestry system. But as this picture shows here from a forest garden, I would say this is not this is the next level up really from agroforestry. There's a very large not only diversity but also complexity, and that then requires a lot of local knowledge about the interactions and managing the interactions between the different species here. Which brings me to our last point, the permaculture. And you see here a permaculture garden a few dozen kilometers from south of Bonn, near Andernach, which is a beautiful space. And it used to be a monocropped field, yeah, with just sugar beet or maize. I can't remember, but that was then turned into a paradise of crop diversity. So permaculture is really making reference to the permanent nature of that. So of that culture. Yeah, so we try to, in permaculture, we try to design systems that are able to provide productivity and resources over a long period of time, that are, in the best meaning of the word, sustainable, yeah, for long periods of time. So here's a definition of permaculture. It's an innovative framework for creating sustainable ways of living. So it's also a practical method of developing ecologically harmonious, efficient, and productive systems that can be used by anyone and anywhere, really. Yeah. So it is, as I said, referring to permanence of that culture, but also because of the high complexity of the interactions, it requires, as I said, a high degree, already for the agroforestry systems, a high degree of local knowledge and a high degree of the interactions between the different partners. In this case of permaculture, normally that requires also a high level of manual labor. And I've been asked many times about research in permaculture systems, and what you realize very quickly is that there are no direct comparison between permaculture, these highly diverse permaculture systems, and monoculture systems, because there's quantitative comparison between these permaculture systems and the monoculture would really make no sense, yeah, because they are so extremely different. So, which brings me back to our one of my first slides. So I think I was able to show that agriculture may actually contribute through diversification to the solution of our many different severe problems that we are facing. And to some degree, I think we can reconcile these aims. There will be still conflicting aims in many cases, but overall, I think diversification offers the possibility to bring these different aims together, but that really requires a thorough transformation of agriculture. We can't just go on like we're doing at the moment. Okay, with that, I'd like to conclude and thank you for your attention, and then I'm happy to take any questions.
Yes, thank you very much, Professor Döring, for this speech and presentation about agroecology and permaculture and agroforestry. So let's start with the discussion. So is there any question in the room? Yes. Sorry?
Just like this, and you repeat it? Or?
Yeah, I repeat.
Yeah, I was curious about the mixing of wheat varieties. I have also downsides or negative effects being observed. And what would you say are the biggest restrictions for farmers to implement?
Okay, as far as I understood, the question is about cultivar mixtures of wheat. Is that correct?
Yeah.
And the potential downsides? Yeah. Okay. So I've wondered that myself many times, really. I give you an example from a different cereal first, which is from barley, malting barley, which has been grown in the former GDR until the fall of the wall in cultivar mixtures in large areas. So really 100,000 hectares in that region, and that was done to restrict the spread of barley powdery mildew and other diseases, because fungicides were very expensive at that time for the former GDR. And that was done really very successfully, reducing the number of sprays substantially, and also reducing the level of disease. And then by the fall of the wall, everything was skipped, the whole system was scrapped, and it was then decided by the new owners of the farms that this is not going to be useful anymore. And of course, the sales of fungicides went up as well. Yeah, so you wonder who benefits from that. So for wheat varieties, you could argue, and that is your question, you could argue that, that has been an argument in the past, I think, many times, that the quality, if you're aiming for baking quality, yeah, the quality is not as reliable. But I think that's actually not the case, because the actual quality that you can measure in the mixture is actually more stable, yeah, over the years, in comparison to a single variety that you grow over several years. I think the point is that millers want to make their mixtures themselves, and they want to have absolute control over which variety they have there, rather than to have a mixture. I think it's a cultural thing. It's not a technical thing, and really, of course, provided that the varieties are maturing at the same time and have, let's say, more or less the same requirements in terms of management. Yeah, otherwise it's really going to be difficult. But there are more than 100 varieties of wheat available, and it's possible. And I think in the UK, for example, there has been the Homegrown Cereals Authority has given out tables which varieties you can actually mix together, yeah, because of their various levels of disease resistance. So they are a bit farther than we are, I think. So I think it's a shift in the value chain that needs to happen, a cultural shift.
Okay, thank you for the answer. So next question.
The new CAP is giving funds to farmers for implementing eco schemes in olive yards. So I was wondering if you know about any example for biodiversity enhancement or either intercropping in olive tree systems.
Sorry, I didn't catch the last bit of the question. Can you repeat, please?
If you know about an example of biodiversity enhancement or either cropping systems, intercropping systems within olive trees.
Within olive trees?
Yes.
No, I don't. I'm aware of studies that look at traditional agroforestry systems, including olive trees, and overall, agroforestry has been shown at that level to increase biodiversity, but there's also a large variability of that effect. So I would think that for olive trees, you would, yeah, I'm not aware of a study, but I'm not an expert on olive growing at all. So sorry.
So next question.
Thank you. I have two questions. I am first, regarding the first question that was asked, curious about the impact of planting a mix of species of wheat on the marketability.
On the what?
Marketability of the wheat mixture. If it is the same for the farmer to sell a mixture of 5 different cultivars of wheat versus a monoculture of the same variety. And second,
I think I've not, sorry, I think I've not understood the key word that you mentioned, the cultural ability.
let me take my mask off.
Yeah.
That probably will help. I'm curious how planting a mixture of species of weed impacts the marketability of the weed. If planting 5 different cultivars would pose a challenge to the farmer to sell it later. Is the question there?
Sorry, the sound quality is not that good. Maybe you can also write on
do you listen?
chat.
Can you hear me?
Yeah, that's better. Yeah.
Yeah. So the question is, if you plant different varieties of weeds, how is the marketability?
The marketability. Okay.
Yes, exactly.
Yeah. Okay. Okay. Thank you. Okay. Yeah, that was the key word that I couldn't understand. Yes, as I said, currently the market is, let's say, in terms of the large value chains, so the large buyers, they're reluctant to take any mixtures of varieties. Yeah, and I'm actually not quite sure why. Why there's really, whether this is a technical issue or whether this could be solved. I mean, the point has been there on the table for at least 30 years. Yeah, so 30-40 years it has been known that variety mixtures are really superior to monocultures in terms of disease restriction and also in terms of yield. So marketability is reduced, but I would say it depends also on the value chain and on who are you selling to. Yeah, because if it's a local bakery or local miller with relatively restricted area that is where he's buying from, or she, then it's actually, I think then it's possible to arrange something that you're providing a mixture of a certain quality, and it's not necessarily so that, yeah, it's a matter of, I would say it's a matter of making arrangements between buyers and producers and buyers. At the global market, perhaps more difficult. But you had the second question.
Thank you for your answer. I'm curious whether these variety mixtures of wheat that you discussed, and also whether the legume and cereal mixtures are already implemented in Germany. If this is something that is more experimental, or if this is something that we could see around your area.
Yes, I would say in comparison to what is actually possible, implementation is very small, but it is being implemented, especially in organic farms or on organic farms. Not every organic farmer grows variety mixtures of cereals, but yes, I would say it's also very difficult to get data for that, how much it's actually implemented. But at least I know of a few farmers who actually do that. Yeah, so I would say adoption is small, but it's present.
Okay, thank you for your answers. Is there any question? So, yeah. So we would like to come to an end. Thank you very much, Professor Döring, for your presentation and for your impactful attendance. So yeah, just to have an overview about the crowd. So I mean, if you haven't seen it before, who's listening actually to you. So I will have a turn with the laptop. Okay, thank you.
Many thanks for the invitation. I will now have to go. I wish you all a very good conference and take care.
Thank you very much.
He should be online already, he said.
Yes, he is online.
Okay, yeah, I talked to him, yeah.
Radina, can you hear me?
Yeah, you're audible.
All right, perfect. I will set up my phone now. I will join the Zoom link that you can use then as camera, and you can spotlight my phone then.
Okay.
All right, perfect.
Okay, then Matthias will share his. Okay. I mean, that works fine. You will talk about text and green faces. Yes. Yeah, no, I will do Photovoltaic and the other one is more Phenotyping. So, but also green faces. Okay. So you gave us the ambitious title. I don't know. Yes. You're not good at this. Yeah, it was somehow provided from the local community. Okay. So we discussed together, but yeah. No, so we do the green face and some stuff that they give video to. I mean, we were with 40 people in yesterday. Okay. For the electrical. Okay. Oh, yeah, that's okay. Okay. Yeah, Oliver showed you around. Oh, okay, perfect. Yes. So some people. Okay, so some videos. I put some videos. So movement of the whole field sake that you use. Yeah. Also, and but the new one is electrical. That's the okay. That's the fun part. Did he move it around also? No. Oh, okay. Yeah. Okay. The connection is perfect. Okay. Okay. So he showed already more stuff. Okay. What else did he say on top about this week? We were in the greenhouses as well. Yeah. And, a short introduction from Anna Lanzaras, in general about the ETG2 Institute. Okay. And we were at the idea. Yeah. And before we were at the EBT Institute in the cellar and saw the whole chamber for the what was it controlling all the factors like CO2, water, wind, etc. And Mr. Bierschenk, Thomas Bierschenk, did some general introduction to the and we had lunch in the sea casino. Okay. I guess it was a quite nice day. Good. Also because of the yeah, many people here. How many people are here? Visiting? Yeah. From all over the world. Yeah. Every country is represented. Nice. And did you finish it?
Radina, can you hear me?
It's not even registered.
Yes.
Oh, okay.
I think you have spotlighted it, right? Is it looking good now?
Looking good. Perfect.
Okay, perfect. Then we can start. If there's something, if there's a problem, you have to send it in the ICEA Bonn chat because my phone now is in use and I can see your chat.
All right. Okay.
Okay, perfect.
Dan yesterday Bibina and Cindy was fighting in the same group. So
Basically, what you can say, you can anytime you say you want to visit Jülich and the name of photos and all that. You join just any or show that you're vaccinated, you don't need a special. You can just go like that. I will also pick up.
Yeah.
She can bring you directly to the gate. So she can be the one inviting you. So she has an or you get at the gate. You can say like, she's there. I'm really here. She's my guest. Okay. That's okay. Okay. Cool. But yeah, that's
That's fine now. Technically.
Now you're busy with this. One more day. But does the online audience also have something, or how is that? They see this. And Thomas is sharing. Yeah. So if we run here, you are instead of front of it, of course. So if we could also put it a bit more this way. Yeah. This way. Yeah. They can see me, and I use this microphone. Yeah, and here's the camera. So yeah, I thought it was the audience. And they need this, or are you sharing also the screen with the presentation?
We can see also the screen.
I mean, now you do your presentation. Yes. First we need Matthias. Okay, and then we need mine. Okay. Matthias will share via Zoom. Yeah. And where's your presentation? This is my presentation. So this is, I mean, so yeah. Oh shit, what I've done. Or is it to go? Okay. Yeah. And then you can ask Matthias to start sharing, because that will be happier. I can ask him also.
Or we write in the chat.
Honestly, I don't know how to.
We can write in the chat.
Yep, you can speak. I mean, they are very good.
Yeah.
I did already. Yes. Perfect. Yeah. It's now in presenter mode, Matthias. Yeah. Good. And I think you can hear me, Matthias. We decided later to transfer this to this computer. But if you have my presentation inside, we can also try that. But probably we will. Let's see.
Yeah, it's better if
Is
you
my
click.
presentation also in there?
Yes. Can you hear me? Can you hear me, Onno?
Yep, we can hear you.
Ah.
Yes, is this your presentation or is it full presentation? Okay, perfect. So yeah, okay. Yeah, let's see. Okay, because here there's some difficulty switching up and down. So if it's all there, let's see if it works. And if I can ask you to click, if you can probably hear me later on. It's not so many. So that's why I said if I ask you click, would that be okay? Okay, perfect. Yep. Okay. This looks good. Matthias will share also my part. Okay.
So we would continue right now at the room is filling. It's nice to see. So for the next presentation, we invited Dr. Onno Muller and Dr. Meier from the Forschungszentrum Jülich. They will talk about the coupling of energy and crop production. The question is synergy to solve use conflicts over land. And they will also talk about phenotyping of plant traits. So I would welcome you, Onno Muller, to the stage.
Thank you. I think the stage is actually Matthias, because Matthias is joining us from online. But I have the pleasure to be here. The second part will be mine, but I think it will be Matthias from online. And we put 2 slides in first because
we've been given an ambitious title by the organizers, the synergy of fields. So if we would like to specify it a bit further, if Matthias can click the next slide. And Matthias, can you hear us? Yeah, exactly. Now you can see Matthias also. Matthias on the left, and my face looks more or less similar to that picture. And what we would like to talk about is, at first, Matthias would like to tell you about photovoltaics and the combination agri-photovoltaics. That's a synergy in the field. And I would like to talk about in the second part about measuring under these photovoltaics, taking the plant perspective. And with that, we go into the real agriculture potentially. How can you use the synergy of fields? But before we go to that synergy, the synergetic use of fields, we would like to go from what do agri-photovoltaics do? How can you phenotype under it? And then we can discuss or open for lunch or coffee afterwards to see further what the synergies may bring us. But that's really an ambitious topic. With that, I think it's first Matthias. Next slide is his online. He will present. And I don't know if the organizer can put Matthias nicely on the screen there as well. Then it's beautiful. Matthias, I cannot hear you yet.
Can you hear me now?
Yeah.
Yes. So a lot of windows popping actually up here. So let me first organize it. You cannot?
We cannot hear you.
You cannot?
We can. The online person can hear you.
We can hear you.
I can hear you.
No, I mean we need to connect it with the speaker. What is the problem? That we could not
hear him.
Hear speak.
Same system maybe.
I think it's better. Yeah, I muted them.
Now.
Now it's okay.
Now you can hear me.
Yes, now we can hear you.
Perfect. Good. So let me minimize the windows there. I also get a message loud and clear from Onno. That's good. So I have several sources of communication here. Okay, so let's start our talk. Onno introduced already. So we will talk about photovoltaics and agri-photovoltaics. So my background is, I'm an electrical engineer, and my background is indeed photovoltaic. So I will explain a bit how the cell works, so from the electrical point of view, and also what agri-photovoltaics means. Trying to click, here it is. So photovoltaics, many of you know what photovoltaics look today. So you have installation of photovoltaic modules on the rooftops, for example, on private houses, but also on grassland, so in the area. This is how photovoltaics in the most cases look today. So we have the modules, as you can see it when you drive through Germany and other countries. And when you look in more detail in these kind of modules, you will see silicon solar cells. So this is a silicon device. You have electrical contacts on the front side, and this is how it looks like when it comes from the fab. And very clean atmosphere here, very clean surface you need to really make the device working. So about 95% of the photovoltaic modules are today made of silicon, but I will also show you later in the talk which other materials we can have. But so integrated photovoltaics of tomorrow might look like that. Here I have a few examples of the integration in vehicles, for example. There's one project also from the research center Jülich, where they try to integrate photovoltaic devices in electric cars. This is an electric car from Aachen, which is produced there. And of course, you can use the electricity of the PV modules to drive some electronics in the car. Not only the engine, but also some other things like air conditioning. So then you also have integrated photovoltaics in buildings. So building integrated photovoltaics, two examples here shown, which is of course nice for the architects, because then you have nice shining facades, and you can play around a bit with the photovoltaic modules. You can make it semi-transparent, here shown on the right-hand side, so that part of the light comes through the PV device and travels into the building, which is also a nice opportunity. And here on the bottom right, you can also see floating photovoltaics where PV cells are mounted or installed on lakes. This is what we call integrated solutions. And of tomorrow, there are some demonstrator projects which are actually running right now. And most probably we will see more in the future. So just briefly looking in the device, how a solar cell looks like. This is a device, so it can be a silicon device, for example, and just want briefly to explain how this in principle works. So on top, we have a negative electrode, and on the bottom, we have a positive electrode. And which is interesting, or which is important here, we have a so-called N-type silicon and P-type silicon. So it means we have, N-type means we have a doped silicon, so that we have more electrons on the top side here, and on the bottom side, P-type means we have a lack of electrons, so means holes in the bottom layer. And in between, we have the interface, and at the interface, we have a so-called space charge region. So where we generate, due to the connection of both layers, where we generate an electric field, so which can drive the electrons into holes. When the light comes now from the top, we can separate electron hole pairs. So, and within the electric fields, the electrons will travel to the bottom, to the top, sorry, and the holes will travel to the bottom. And with that traveling, we have electricity going through, lamps, for example, or something else. This is how a silicon device in principle works. So we have two layers of silicon, which is doped positive or negative. What is the potential of photovoltaics in the installation? This is the question what we are asking ourselves, and especially looking at the agricultural sector, what can we do? Because nowadays we are discussing in Germany also to bring the Energiewende a bit more forward, but then we see the roofs will not work to cover the whole potential or the needs, so to say, what we need in the future. So here we have one source of the German Mirror article, which nicely shows what is the potential of photovoltaics in the future and what we can do. Now on the left-hand side, you see the requirements in 2040 in Germany, what you would need to hold the Paris climate targets. So to really keep them, we need a photovoltaic installation of 300 to 450 gigawatt. Actually, nowadays, we are in Germany at 50 gigawatts. So it's a huge task to really install more and more. And here on the right-hand side, we have different sectors. I would just briefly look into that, which kind of potentials we have. So on the right-hand side, we have for buildings and roof installation of photovoltaics about 1,000 gigawatts to be installed. So it sounds like a very huge potential if every roof is covered in Germany. But of course, you can imagine roofs, not only roofs, but also facades. But you can imagine that also some regulations and so rental houses and so on, which can block these installations in the future. And this is actually the case in Germany. So the regulations are not so clear. There are different business models to drive the photovoltaic devices on the roof house. So we will not have 1,000 gigawatts installed on the rooftops in Germany. And then we have other potentials, installations on roads, parking places, floating PV, vehicle integrated PV. And so just to show you a bit numbers on that side. And on the left-hand side, you see the big potential of installations on agricultural land. Means fields to produce corn, berries, fruits, wine, grassland, and so on and so on. And this potential is about 1,700 gigawatts, which is of course huge. So to keep the potential, we are of course interested also to keep the fields and the agricultural use. Otherwise, we will have a problem with the food production in Germany. And one solution is to install agri-photovoltaic system. So here are some systems shown how agri-photovoltaics can look like. On the left-hand side, one system of Fraunhofer ISE, one of our partners, and which is a quite huge system, so that the harvester can drive under it to harvest corn, for example, or wheat. And on the right-hand side, it's a bit lower system where you grow raspberries And this is installed in the Netherlands by Biodiverse. It's an energy company in Germany. And this is what I said before, a bit lower system, which of course the installation cost is a bit lower. And yeah, so we visited the plant once, and it seems that you really get nice fruits and nice quality of fruit. So it's working quite good. Then we have vertical system where you can drive through the panels and harvest some grass. And here on the bottom right, you have also greenhouse installations, where I will talk about a bit on my slide. So these are the different kind of agri-photovoltaics. So our institute is also working in the topic, as you can imagine. So we started to work together with a company called Sun Farming. They have a research park in Brandenburg. So it's in the west of Germany. It looks like that from the top. And we planted some ornamental plants here under these kind of panels to try out how the growth performs in shaded, non-shaded conditions. And what we did is we have image analysis. So we installed some cameras, and Onno will also talk a bit more in detail how it looks like. So we installed cameras to take original pictures. Here is one example of sugar beet, which was just the first example and just first try for the student to convert this picture into black-white analysis pictures, to count the pixels here on of the white leaves. And then we can also make a control image to compare if the leaf area is similar to the one of the original picture. And this is what we did. So, and of course we counted the pixels and how much green, and you know, I'm an electrical engineer, and Onno can explain in more detail a bit better about that, but how much green we will have under the panels. So this is the installation, how it looked like. So we did it last summer. This is the system, and here's the installation of the camera of our postdoc, Christoph, and our student, Surab, who installed the cameras under the PV panels. Here's also a nice view through the PV panel, so you can see it's not totally shaded. You have a bit light coming through, but of course, if you look on the left-hand side here, we have, of course, a huge shaded area. And here the ornamental plants, which are arranged, and we had several pots where ornamental plants are placed. And this is one example what we generated in Rathenau in the system. You can see here is the time axis, and here on the y-axis is the projected leaf area recorded by the camera. And here we have two cases. On the one hand side, we have the glass. So the glass is the open area, so to say, where light can come through. And we have the PV panels, which is shaded. And you can see, so the main message here at that point is we can measure a difference between the ornamental plants placed in the shade and also in the light region, which is for you, of course, not very surprising. But for us, of course, it's important so that we can measure the leaf area also within the system, and that we can measure some differences, which of course has influence on the quality of the biomass, etc., etc. So you can okay, a bit. What you can ask why the curve is going down here. Here the flowering starts, so you could not nicely measure the leaf area from the plant. So this is why at that point the images are a bit disturbed from the flowers. On the right-hand side, you see an example of the total dry mass measurements over the pot number. And here you can also see shaded is orange, open is blue, that we can also measure difference in the biomass within the system. But Onno will explain more details on that. What we can also see is results from our partner, Fraunhofer Institut ISE. The system is a different system, or what I've shown before, and they measured the growth of wheat, potato, celery, clover grass, and here you have the crop yield in percent compared to the reference area. Here you have the experiment. Here's the growth under the panels, and here you have a reference area, and you can nicely see that you have differences in the yield in 2 different years, 2017 and 2018. In 2017, you have less crop yield. In 2018, you have a bit higher crop yield. This is because in 2018, the year and the climate was a bit drier and very hot climate. So what we can see here that also the photovoltaics modules can protect the plants from extreme heat, from extreme sun, and also from evaporation of the water underneath. This is one example of Fraunhofer ISO. Now I'm running a bit out of time. So the last slides are showing our system, what we have installed end of last year in close to Jülich in Morschenich. It's a brown coal area. You can see the digging area in the back and also the power plants which are burning the coal. And in the front, we have our new agri-photovoltaic system installed in Morschenich. And here we have a top view. So we have different kind of systems. Starting on the left-hand side, we have a south-oriented agri-photovoltaic system. So it's looking so PV panels are looking into the south. And here we are testing different kind of roof combinations. In between the PV panels, here we have some plastic roof, which is transparent. Here we have some hail nets installed. And so we are trying a bit what is a good roof configuration. Here we have our reference area. Onno will also explain why we have this nice metal installation here. And this is the other south-oriented system where we don't have roofs in between here. So these are open to have also reference to the roof area, again, a reference area. And here in the back, we have so-called future tracking agri-photovoltaic system. It means tracking means we are tracking the sun by moving the PV panels when the sun is moving. So we follow the sun with the PV panels. This is one thing, so we can increase also generation of electricity with these two systems in the back. On the one hand, we can also control the moving PV panels to protect the plants in different growth conditions or also from extreme weather, for example. So if we make it flat, for example, we can protect the plants against hail and extreme rain and so on. This is the idea. Here's the system a bit in more detail. So this is a tracking system. Here on the right-hand side, you can see how it can be moved from left to right. And yeah, what I said before, we can control it. Shift a bit the window here. We can collect rainwater. Here's a gutter in the middle of the PV panels to collect the rainwater and then use it for irrigation under the PV panels. And yeah, the automated phenotyping system, Onno will explain in more detail how it looks like these kind of systems. Maybe the last slide or one of the last slides is to show also the potential of the technology of agri-photovoltaics. Here's one installation shown in Korea, built by a Chinese company, and this installation has about 35 megawatts. So it means we have 35 hectares about where this agri-photovoltaic system is installed. So the potential of scaling, for example, is given with this technology. Okay, this is the last slide I want to show you. It's not only making PV panels on top of fields, but also integrating of photovoltaic systems and photovoltaic modules in greenhouse installations. This is one proposal we are actually writing on our knee to study and to build a demonstrator on European level with our European partners, how to integrate photovoltaics in greenhouses. They should be, of course, semi-transparent so that a part of the light travels through the glass, the greenhouse, to make plant growth possible. On the right-hand side, you have an example of a spectrum. So the red curve is the transparency of the PV panels, and here in blue you have the chlorophyll A absorption spectrum, and you can see there is a nice fit between these two spectrum. That for the peaks, the chlorophyll needs the absorption or need the transparency. The PV panels are transparent, but there is a lot of things to study in the future, and this will be done in hopefully successful European project. And with that, Onno, I would like to switch to your presentation, and the questions most probably will be asked later.
Thanks a lot, Matthias. Maybe we try that Matthias just continues clicking. And yes. Okay, thanks. Yeah, this slide. Matthias introduced the photovoltaics. and I would like to continue on measuring plants. And I put this field under the photovoltaics, where you see what happens under photovoltaics, shade and sun. And you can imagine that plants respond to that. Matthias showed you the yield of a potato over 2 years, where it was largely different, one time positive, one time 10% less. That's a really crude scale, yield, and in the end we're all interested in the yield, but I would like to step one step in more detail. Can we grow plants that are adapted to these shade conditions? Or how do plants respond to the shade and the sun under the panels? Or what is the potential of growing plants in this variety of climates, and how can we quantify that? Similar questions also would derive from the talk from Thomas Döring earlier, when you have different diversity measures. What is the potential of such a diversity measure? Can we quantify it? We can quantify it at the end of the yield, but the yield is composed of a lot of different parameters. And I would like to show you some parameters that we, as with a field phenotyping technologies, we try to quantify. You were, I understood, yesterday in Forschung Centrum Jülich. That's also where both Matthias and I am from. And you may have seen some parts, and I would have loved to show those in the field, but I brought you some movie also to get an impression how these big machines are moving. Next slide, please, Matthias. Yeah. Yes, this was the extension to the field. I think all of you know already this was the field under the PV. On the left side, the situation in Morschenich, close to Jülich. On the right side, in Hegelbach, somewhere middle Germany. I get a nice laser pointer from Matthias. On the right side in Germany, but the point is the shade. To see the different shades, and of course this is a nice potential agricultural approach, but it's really crude. I think, or Matthias and I, we are calling our APV system also agriculture horticultural. This is agriculture, say we put systems on the ground and we continue with large-scale agriculture, but it needs a lot of infrastructure. You need to put the system so high, 4 meters, that all the tractors can drive through. If you could reduce the system or adapt machines under it, you may come at a lower cost and it may be easier compatible for the future. That's other examples he showed, for example, with berries, and examples in the Netherlands where the systems were lower, adapted for plant growth. But key of my talk is about quantifying plant rates. And can I have the next slide, please? Quantifying plant rates is something, as you've seen me doing. No, the video, please, Matthias. Could you run the video? Yeah, and that should start. And this is where, yes, this is where there it's working and here it's not working. Would it work to play the video, Matthias?
Yeah, where I have to click actually.
It should go start automatically, and otherwise in the low, if you go to the lower end of the slide, then it should you could maybe start it.
Oh, yeah.
Yeah.
Yeah, maybe the laser pointer is now. Oh, Zeigeroption. Let me see. Why I can. I can. Yeah.
Oh yeah. Yeah.
Yes. Yeah.
Oh, perfect. Sorry, but thanks, Matthias. But what I what's shown here is that if we quantify plant rates, diversity are on the panels that and over seasons, it's continuously changing. And that's what we want to quantify to come to the final yield. And it's stuck here now. Is it moving at your side, Matthias?
No.
Okay, I'm gonna change. That's easier. Otherwise,
No. Look. Oh, no. Now.
Yeah. Okay. But we go over the season. As I said, it's otherwise it's over the seasons. It's changeable. And how do we quantify it? We also choose not to be static on one point, but we use one of the machines that you've seen, for example, yesterday on the field. They can move flexible over the field. We have the drones, but it doesn't replace the manual measurements, as we here saw bikes pushing through the fields. I think Jan or Philip has been doing that as well. They've been walking through the fields, putting the sensors. We have the automation to get to higher throughput over the seasons. Like here you see a preset of the one you saw yesterday, moving over fields from winter to spring to summertime. And the advantage is this platform, it can precision in centimeter accuracies. So we can measure the same crop over and over on time and time in different plots over days in different crops. For example, here in potato, in wheat, or in also it can rise high enough that you can measure over maize, about 3 meter highs we put. And we also can put it, we want to quantify traits not only in the plants that they grow nowadays, but also on the future conditions. And that's where this last part of the picture is. You see again an overview of the field. In the middle, you see that big bridge, as I said, is made big because we can, with limited invasion, we don't touch the plants that are grown under. We can go to each point and we have a sensor platform that we can move in centimeters accuracy and in different heights, any time of day. And the system behind it, you see here what we call is a ring or CO2 ring. We call it our breed phase. That's a ring that blows CO2 in the field. And because it's blowing CO2 in the field, we can investigate how plants respond to these future growth conditions. In the future, there will be higher CO2 conditions, and we would like to quantify plant traits when they're growing there. Our general concept, to put it a bit more structured than just a video like this. Next slide, please, Matthias. Next slide.
Yes, yes. Clicking. Why? Yeah.
Yeah, thank you. Okay, no more videos, but more static and a bit more structure. If I talk about field phenotyping, or if I said about the introduction either on the PV or in diversity panels, to quantify plant traits in the field, how do we go about? What is our concept behind it? I think it's a concept you may all be familiar with, but nevertheless, I would like to repeat it again. If you talk about phenotyping, but also talk about general agriculture, there's always the interaction with the environment. And nowadays, more and more also in prediction models, the environment needs to be quantified. If you can quantify the environment, you know about half of how the plants will respond. Often it's forgotten. You use an environment station far away. Oh, we have an environment station stand there, and that's the data used. But you would like to know of your field, your area. In experiments with different varieties, you want to actually know, would like to know the environment in one single plot. Because if you have a deep rooting genotype, it may have a different water holding than a shallow rooting genotype. So that's on the right side, environmental sensors. I must admit, we ourselves do not invest enough in it yet. If you talk about phenotyping, it's quantifying the genotype times the environment. So environment is equally important as the genotype and as the phenotyping itself. But you will notice if we talk about phenotyping, mostly we focus on how do we quantify the plant. That's the other 4 pillars of this concept. How do we quantify the plant? How do you quantify the plant traits? Different plant traits. Traits which I split up a bit more in detail than yield. For example, roots, structure, water, or photosynthesis. All of them contribute to the yield in the end, but we cannot quantify them all equally. Below ground is rather difficult still at the moment in the field. In Jülich, you have seen some systems, the different rhizotrons that you could see in a greenhouse below ground, or the MRI you may have seen there, where you can, with magnetic resonance images, you can look below ground, but in the field, we're not so far. Above ground, we're pretty far. And I will show you some examples, for example, measuring photosynthesis in the field. How do we do it? The sensors. We in Jülich develop several sensors, and we work together with the different companies to have the right sensors to measure, for example, photosynthesis in the field. Often we use their fluorescence, the process of when light comes upon a leaf, sugars are made, and when the light is observed, fluorescence, light is reflected, but also fluorescence comes from the leaf. This is what we can measure with different sensors. And to have these sensors at the right place, we need different positioning systems. And of course, there are the different experiments where everything comes together, where the sensors, the positioning systems come together to measure the traits under different environmental conditions. 5 pillars of the field phenotyping concept as we see it. Next slide, please. Matthias, next slide, please. Yeah. Starting with the first in the middle, the pillar is positioning systems. You've seen some, and although it's just a technical part, and I think also the positioning systems, they should follow the sensors and the traits that you want to measure. So that's why we also have the luxury or to have a wide range of positioning systems. And it's not that I say we really need sensor, the electric moving GPS here system that you saw yesterday in front of our institute. If we can measure the same way with a bike, or if we can measure it better with a bike that is pushed by hand, we will do it by hand, because in the end we want to measure how, for example, this lettuce is growing over time in this field. But I must admit, it's easier and it's it's also sometimes fun to fly with the drone over it, or with a fixed wing, or to have these large bridges measuring over, or this little tractor giving a lot of diesel smell. But anyhow, you can repeatedly, easily, without pulling up this sensor anytime again, you can use it. So in short, the positioning sensors, they make sense if we use them in functional throughput. So you decide beforehand how often you want to measure and where you want to measure and at what time points, at relevant outputs. Then we have positioning systems are really useful for plant phenotyping. Next slide, please. Yep. I cannot talk about all the traits with you. I would like to talk about some traits, and of course some traits that myself, and also in Jülich, we have quite some expertise in. And one of these traits is photosynthesis. If you talk to a real photosynthesis guy, I'm a half photosynthesis guy, you would say photosynthesis is the key of all processes. I think it's also true. But in evolution, it's one of the primary processes. All the plants have chlorophyll and they photosynthesize. You may wonder, after 20 years of breeding, or actually genetic modification also sometimes, they didn't really manage to change this basic process of photosynthesis. That is still the same. But it's all the factors around it, structure of plants, orientation of leaves. That's what has changed and improved crop production a lot. Nevertheless, photosynthesis is indeed still the key and the core process of getting the sugars or getting to your crop production. So quantifying it is essential and rather interesting as well. And as such, we have also quantifying photosynthesis at different levels. We do that at the leaf level. We also recognize that plants don't grow only as one leaf, they grow in plots. So in Campus Klein-Altendorf that you may have visited, you see, for example, containers where we can grow plants in canopies, or we have it, we measure in the field in bigger plots, and there we have, for example, an instrument called a light-induced fluorescent transient instrument that can measure all those plots. And these are sensors also on this range, handheld or canopy, are pretty heavy, but we can put them with different positioning systems. If you go lighter, you can measure also from drones with cameras, measuring sun-induced fluorescence, or you can put a more heavy sensor in an airplane, that you can measure larger regions. And the outlook for the actually next year will be, there will be a satellite also launched, led by Rascher from our institute mostly, that will be measuring sun-induced fluorescence from the sky at a much larger scale, at about 300 meters. So you may wonder if that's still phenotyping, that you want to identify different genotypes, but certainly for agriculture or biodiversity, other characteristics, a very important trait. Besides from leaf to almost large scale field level, there's another separation between measuring photosynthesis, and that's active and passive. Active means you send a light to the plant and you retrieve it back. And with that, you can quantify photosynthesis pretty precisely. Passive means you use the sunlight, and the sunlight just shines on the plant and you measure the fluorescence. You need to measure precisely where this fluorescence window is, these few wavelengths that are fluorescence. If you ask me in the break, I cannot tell you more detail which wavelengths exactly you would like to know. And these three different methods are important to distinguish, because active light you can measure, you can only send it from proximal sensing, and remote sensing will be more sun-induced fluorescence sensing, more from a larger distance. That's when it goes to the satellite. Okay, short introduction about photosynthesis from different skills. Next slide, please. Next 4 slides or something, and then I come back to the PV. Are some examples of using photosynthesis and other traits in current experiments, one of the pillars of experiments, which I thought that could interest you. One of them is drought. Drought, a prominent topic for all of you. How do plants respond to it? In this case, we said we don't wait for a natural drought to occur, because we cannot easily quantify it. In this experiment, we went to Maricopa in the USA, in the desert, and we irrigated the crops in the desert in the winter. And that's why you see this green field and brown around it. And what we are interested in, we work together with in Döring wheat, about 200 varieties. How do they respond to this drought? And of course, our part, we brought a sensor that can measure fluorescence, that can measure photosynthesis. And all the details you can find in this publication, but I want to share you one. If you stop giving water in the desert, on the y-axis and the x-axis, you see the time after day 0 when they stop giving water. On the y-axis, the percentage change in photosynthetic efficiency and some other parameters. And what you see, it decreases. As will yield decrease and water content will decrease, but this is a parameter we can measure on top of the plants. What you also see, it's a straight line, but you see kind of, if this is the 0, there's first a funny increase in photosynthesis. There's quite some dynamic. So there's a quick response already one day after you stop giving water, plants response in photosynthesis going a little bit up and then later going down. So these dynamics may also be very early indicators that you cannot see directly the soil water content. Soil water content goes also down, but not as dynamic as photosynthesis down. So here we could show, and we could also separate genotypes that are very responsive to drought and others that are not. Next slide, please. Now I talked about direct responses to water at very short term, or I even said maybe earlier. I would like to detect it earlier. Another thing is about photosynthesis, and that's how photosynthesis or phenotyping is often used. Another way that we explored in this paper, and I think is also relevant if you think about under PV, different shade, different light over time. Thomas Döring addressed it as the sun hours over the time of a growing season. It's that the plant is not growing at one time point, it's growing over a whole season. If we could quantify this photosynthesis, if you would know how much photosynthesis the plant do over their lifetime, this should then theoretically correlate precisely with their yield or with their biomass. A lot of attempts have been done, and a lot of attempts have also been modeling this, and there was not so much, there was a lot of variation there. So what we said now, we have the machines, we have the positioning systems, and we can do that very accurately and very precisely. So we measured for several months, this is May data, but we measure accurately every time, every minute photosynthesis. And this we extrapolated, or we said cumulative, we cumulated these effects in response to light and photosynthesis and how efficient it was. And if you would use all these measurements, direct measurements of photosynthesis, every minute or every hour in the greenhouses, in the field, then if you put that into your model, then you have a pretty high correlation or in relation to biomass of these plants. So the amount of biomass could in this case be explained by the variation in photosynthesis. May sound very trivial, but so far studies have not been able to show this because there's so many other factors that determine photosynthesis. So we were very happy with this result. Next slide, please. I said already many other factors determine photosynthesis. So I can say it, but I also would like to show something about it. Otherwise, I'm just saying many other factors. And two slides, two projects that we're working on these many other factors, and also the importance of quantifying structure or just quantifying shape. Even we called one project on the right shape and color, because from shape and color we can derive already so many relevant parameters for a breeder or for a grower. For example, the difference on the right side you see in carrot leaves from green to yellow. If we Dirk Jolet, a PhD student with us, he quantified it with a nice color reference, and he could with that identify very clearly the disease infection Artenaria, I thought it was in carrot. On the left side, using a drone, another more close to practice question was cauliflowers. We harvest them, we walk over the field in about a week time, and then we check every time what is the right size of the cauliflower. Could you tell us when the right size of the cauliflower is there? So we imaged these cauliflowers, and we made together with Ribana Rascher and Jana Kierdor of the University of Bonn, we made a machine learning model. We used these images and said, okay, can we now retrospectively, what we know, what is the right size of the cauliflower, can we find out which cauliflowers are really ripe or have the right size to harvest? A project called Optico. And we got pretty far there, but still rather difficult, but with machine learning methods, they could identify or predict roughly when this cauliflower had the right size. Next slide, please. Yeah, this is International Conference of Youth Agriculture. So the examples I just showed are only in Germany. And I think, yeah, nice. We have a nice agricultural landscape, but I think agriculture happens only for a small part in Germany. And as such, I think such a kind of studies are equally or even sometimes more exciting that we're doing. And I would like to show you one example where we bring our phenotyping technologies to a cassava field. Cassava, major crop in South America, Africa. And we work here together with the Bill and Melinda Gates Foundation. And again, we bring our drones and we quantify over time the plant canopies. And from the plant canopies, you could arrive from these pictures, for example, you could derive the plant height. And here you see it after days of planting. And these different growth parameters of plant height are then again compared to different genotypes that we had with different sink source ratios that are actually by other partners genetically modified to improve the yield. It is a different aspect where they genetically modify photosynthesis. Here they go for sink source for the whole plant perspective. And so far, early results show that this may indeed increase the yield of cassava and maybe a promising road forward to bring the varieties, these new varieties on the field. Next slide, please. New varieties on the field for the future. This I touched already about in the beginning, is on the elevated CO2. What is the future? The future is not as it looks today. For sure, we know that CO2 will be increasing. CO2 is increasing. We are at 400 ppm, but you will remember that as the start of your studies. When I started with my studies, it was about 350 ppm, or actually 320 if I'm honest. But my measurements, when I first started measuring photosynthesis, I had to put in 350 ppm. 50 ppm difference. In a few years times, it will be 600 ppm. That's the conditions we measured at. CO2 in the air, directly taken up by plants, directly increase in production. About 20% higher, have studies shown, like this one. Can you click a few times, Matthias? Where they put pipes in the system, in the field, and they blow CO2. We are not a, could you click, Matthias? We're not the only system, but these pipes, they blow a CO2 in it, and these are existing worldwide. About 20 years ago, they had about several systems. That this year's a few are remaining in Nanjing, in Illinois, in the United States, in Japan, some with rice, but that is still blowing CO2. Pretty costly to get the CO2 in, but also interesting that we get the results. And I would like to share you some results of a study that we did here. Next slide, please. And that is soybeans. We grew soybeans. It's at the northern region of where they can grow. And we grow, this is again the same ring in Campus Kanaldorf. You see soybeans are still green when the rest was harvested. And we also showed for soybeans a pretty high increase, 46%. It's really high. So if you grow plants on the high CO2, they had much higher yield because there's so much more carbon available. But if you then look in more detail and this increase in soybeans, does the quantity increases 46, but does the quality remains the same? And that was not the case. If you then look in the mean concentration of different nutrients in these plants, you see overall all these nutrients decrease in quality. So you get more carbon in the crop, but you don't net equally increase in quality. So you may wonder, you may ask, how can we breed them for plants or crops that maintain their quality? Do we have to give more fertilizer, or do we have different uptake mechanisms? So these are questions that will come after. Next slide, please. These are also the same questions that we were trying to answer, and Oliver Knopf showed you around yesterday, I understood. And he's trying to answer these questions in a larger project for winter wheat. So he's been growing winter wheat now. This year will be the second year that he grows a population of the German breeders. And these are the data of last year, where you also see there's about 20% more straw biomass at harvest, a bit less in yield of different varieties. You see also some variation between the varieties, and the yield increased about 7% on average. Some varieties 20%, some varieties didn't increase yield. And all of it measures the photosynthesis in this plot. And if you are interested or to follow this experiment, You could look up by Google, for example, PhenoCam and breed phase, and you can see the live pictures of this experiment, how it's growing over time. And of course, questions you can ask Olaf or of me. Next slide, please. Last slide, back to the beginning, back to the photovoltaics as Matthias started with, and he said, oh, Onno will talk a lot about it. I didn't talk so much about photovoltaics because this is the situation at the moment. We just installed it, but the idea is, or the concept is also that all this field phenotyping talk that I did, the same concept, the same quantifying of plants, the same questions of how does shade vary the plant growth as we do with CO2, we will answer, or attempt to answer under this system. That's indeed why we have also open systems or racks standing there, because we thought it's not easy to drive through under these panels. So we designed another positioning system based upon the rails that we will hang from these roofs underneath, and so easily a little bit like a train, but then upside down. It can carry our sensors and measure frequently over time. This at the moment they're installing it. I mean, these parts are there. Next week the rails will come, and then more importantly the plants have to come because it's pretty brown yet. Studies that we will do are some previous studies we have done, like we did here in north, close to Berlin. There we just put plants under it, and we just imaged the plants, and we showed that there were differences between shaded and some plants in growth. Rather obvious. And the question in all these parts will be, what are the effects of shading or partial shading on the crop performance at different levels, measuring from structure, photosynthesis, and also yield in the end. Other small experiment as last teaser is what Matthias showed, the photovoltaic, when you can use panels that don't transmit any light, but you could think of the future also of panels that can transmit the light that plants can still grow. Organic photovoltaics have these capacities. Here you can see some panels we put above spinach plants in the greenhouse. They look reddish, bluish, but they let about 20 to 30% of the light through. And in winter time, we could still grow plants under it. Of course, there comes also light from the sides, but that would be similar if you would disperse them over the greenhouse. So this may be an outlook to the future, how it may occur, and it may end up, if the price, etc., end up also in agriculture fields. With that, I would like to leave you. The next slide is the final slide, I think. Yep. And thank you for your attention. Open for any questions. I will be here for lunch, coffee, and thank you. And Matthias equally is online to answer questions. Yeah.
Yes. Is it working?
I hope so.
I mean, no, so yeah, thank you very much, Dr. Onno Muller and Dr. Matthias Meier, for this impactful presentation about agri-photovoltaics and phenotyping in plant traits. Yeah, are there any questions to the speaker? I see a lot of hands.
I would like to ask, you're using photovoltaics in a part of the world where light is one of the biggest limitations for plant growth. Wouldn't it be more interesting to use photovoltaics like in the tropics, where light intensity is less of the limiting factor?
Yes. Can be very short. The question of more is always difficult. I mean, photovoltaics here work also, and you're correct that part of the season light is limited in here. And so we have, if you have photovoltaics on your house or whatever, you will see in summer you have it's the inverse of, it is directly corresponding to the sun hours graph that Thomas Döring was showing. So it's actually, you see also if you look at greenhouses maybe around here, you see they paint them white. There's too much light also at a certain time of the year. So it's not fully true that's you're right. There's way more potential in other areas than here, but that doesn't mean that there's no potential in these areas. Yeah, but you're correct. Yeah.
I'm just wondering whether we can exploit these photovoltaics to have more light for the plants. I don't know if it makes sense to have light in the field, but
Yeah. No, maybe, yeah, Matthias, but I also, yeah, Matthias also there to answer, but I think, Katja, you raise an interesting question because we have, when we were discussing about we are discussing about this organic photovoltaics, and there the question is in it, how much light does a plant need? Which window of light does it still need? And talking to the growers, they actually said, yeah, but wait a moment, why do you make your life so difficult? Because if you would put a photovoltaic cells on top, I, with the energy that produces, could calculate, and he was a grower who came from the vertical farming direction. He said, I could power so many LEDs and produce so many lettuce or crops that can grow on the LED lighting. And that would be, if you look at service areas, his estimate was a quick calculation that he would grow 1.5 to 2 times the area over the whole year than compared to normal light conditions. So your question, if you would put the photovoltaic cell completely dark and connect it to LED lights, there should be a battery in between, so continuous lighting. According to his calculations of this grower, there could be more production than you would just use normal light during the normal day. Yeah, but I don't have the numbers here, but that is what a quick calculation we do. And of course, there are many there's a lot of advantages of doing this under very controlled conditions, but there are a lot of downsides also on growing plants in these structures and all the isolation and stuff you need to grow plants like that. But there's a certain sector or certain plants that are grown in greenhouse already that may be suitable for that.
Oh, I have a bit needs of understanding. You said that you're using CO2 in Agroforestry. Yeah, please, can you clarify because CO2 is abundant?
Certainly. I showed you a ring, and this ring blows CO2 in the field, and it does that to, and I didn't clearly explain it, to increase the levels to 600 to simulate a future CO2 condition. So it is an experiment where we want to simulate the condition of about 50 to 100 years later. So, and because CO2 is so important for plant growth. So that's indeed, it's just blowing CO2 in the air, which is abundant. And I don't know if you're hinting at that, but you're pointing out indeed an abundant parameter. As CO2 was, if you look at the response to photosynthesis and CO2, it's linear and then saturating. And this point is approaching more and more. So we were at 300 ambient, was somewhere here. And now we're already at 400 ambient. So we're more and more getting in this saturating part, or in other words, in the abundant part of photosynthesis, where CO2 doesn't matter anymore. It's not a limiting factor anymore. So, and that's maybe happening now. So the results I see that there's only some small increase in yield compared to studies that were done 20 years ago. Maybe indeed that it's getting closer to where CO2 is abundant already on the current conditions.
The second question,
yeah.
What is the height of CO2 machine that you have in the field? From which height you are blowing the CO2?
Yeah, CO2 is blown. It blows at the height of top of the canopy, and it increases with height when the canopy grows. Yeah.
Further questions? Just test the microphone.
Hi. I
Hi.
have a question about the use of photovoltaics and transmit light. If we use that, does that reduce the efficiency of the solar panels?
No. That's the challenge. Yeah, I mean they're competing for the same light, so and that's the efficiency decreases the more light you let transmit, the less efficient the solar panels get. And that's the balance. That's the project Matthias was referring to. That's the balance we're searching for. What is the right efficiency to still be photosynthetic productive under it, but also be productive for the energy of the panels, so that it's worth the cost to invest in it. Yeah.
Thank you so much for your presentation. I think this topic is so interesting and it's somewhat new for me. I'm more familiar with photovoltaics on free land, and I understand that one of the biggest challenges with the management is actually how you manage the crop or green or rocks in some cases underneath. And a lot of concern is that you back up the mower or you back up the tractor and then you ruin millions of dollars worth of equipment, right? So I'm really curious how farmers are talking with you about this technology and also their effect on land prices, because that's one of the things that happens also with the photovoltaic on pre-agricultural land is drives land prices. So I'm just curious on your insight on the topic.
Yeah, I think I let this answer by Matthias. Could you hear it, Matthias?
Yeah, I could hear it partly. So the question, maybe I can. So maybe you can briefly summarize, Onno, so that I can.
Yeah. So the question is, if you talk about photovoltaics, the lady in the room is experienced with photovoltaics, and she said in her experience, it's the interaction with the farmers driving under or managing the land, also if it's just on normal bare land or rocks, is the trick, because you are in danger of one wrong move of the tractor of driving down millions of equipment under it. And how do we deal with that, or how is our interaction with farmers about driving under these, cultivating crops under the panels? And the second question was, how is it with land prices? Because as soon as you put photovoltaics on this, land prices increase. So you have an interesting influence on the market in terms of land prices.
Yeah. So the first question about yeah, okay. So the first question about how we deal with that and how we talk to the farmers. I mean, this is exactly what we want to find out here in our demonstration system. Especially the picture what you see here. We have a height of, so drive-through height of 2.50 meters, and the poles are like 4 meters wide. So it's of course a challenge to drive. Yeah, exactly. This is of course a challenge for the farmers, but in our system, as Onno mentioned, we are calling that Agri and Horti PV system. So we will more concentrate on berries and fruits and maybe some salad production of tomato, where, so what we think, and also for our region, where a bit more handworks is needed. Handworks means so small machinery driving through that, not fully automated. So what we think is, and when we talk to our colleagues and to the farmers in the region, they don't see really megawatt parks where you can drive under it with a big harvester with GPS control. This is what we do not see talking to the farmers. We see more like the system what we see here. So between 1 and 2 hectares, where we have horticultural use underneath, where we have a lot of hand pickings and so on. There we can see, or we will see the first photovoltaic system. This is also, so my thinking and also my feeling when we talk to the farmers. But of course, these are now in Germany the first steps, and we have to show and we have to demonstrate how agriculture use or horticulture use in these kind of systems can work in principle. This is the challenge of the next years. And the second question was about the pricing. Yes, this is quite interesting. The price of the land might increase. This is not very sure at the moment, I would say. So agri-photovoltaics systems will be supported by our government in the next years. This is what Minister Habeck mentioned. We will have an Easter package with some regulations and so on to support agri-photovoltaic systems. And within that system package, so about the regulations, we will also see how to deal with different land prices and so on. And also the EU funding, the Agrar funding of the EU, how to deal with that when we put agri or photovoltaics on agriculture land. So yes, this can happen, of course, but there are also business models of energy companies paying exactly for that. So they will pay your rent for installing the energy system on your field, and they will deal with all the costs also on the energy market and so on. There are business models exactly about that. So yeah.
Did it answer your questions? Yeah.
Maybe,
yeah, but
maybe one more comment, because now you're also talking in the round a bit about, okay, how efficient and how much light will come through. The motivation really to install agri-photovoltaic system is what I've shown before, the potential and the needs of photovoltaic, what we will have in 2040, for example. So what I've shown, some scenarios are calculating with 400, 500, maybe up to 600 gigawatt we have to install, not only in Germany, but all over the world. And so the roofs will not be enough. So we have to go on the area and onto the fields, and to keep the land for agriculture use, we have to combine both. So, and this we have to find out. So of course, we will have a reduction in efficiency and also in land coverage, area coverage of the PV, but to find the right balance of energy production, also agricultural use and biomass production, this is the topic and the aim, because PV will go onto the fields, definitely. And to have because we need the areas, and to keep really the agricultural land and use underneath, we want to go in that direction. Maybe, yeah.
Thanks. Thank you very much. So maybe one more question, one more last question, then we are. Okay, shout it.
Yeah. So if I remember right, in my class in university, plants don't need to be light. So is it possible to make photovoltaics that work on green lights and that through like red and blue lights so that plants can grow almost optimally? And is it something you are already doing?
We're not already doing it, but that's exactly what we are now discussing in this project. And there are some projects also in the Netherlands, in the TU Eindhoven, I saw, where they're exactly looking at this cutoff of these wavelengths. You're right, green light, because it reflects, it's less taken up by plants. So could you there have a lower transmittance? In other words, harvest more in the photovoltaics? Yeah, that's exactly how we. These are steps of fine-tuning that are have to go together with organic compounds in the photovoltaics. How much can you fine-tune them? Then we're talking to the technician guys, and in other institutes we're really specialists in these photovoltaic cells. But to what extent can they really pinpoint specific? How can they find organic materials that can filter out these wavelengths? But that would be the next steps. We first do our proposal is first to do it broadly. Say there's the so-called photosynthetic active radiation for 400 to 800 nanometer roughly. If we would have this transmittable and the rest for photovoltaics, how much would that be in terms of efficiency? Your question earlier. How much would be left over for the photovoltaic cells? The next step will be indeed finding these regions, green light, smaller wavelengths, to filter those out. Yeah, remember it well from university.
Okay. Yeah, then unfortunately we need to come to an end for this impactful presentation. Thank you very much, Dr. Onno Muller, Dr. Matthias. Yeah, now we have a coffee break until 11:15. So you are free to drink coffee, to get in contact with the speaker. Yes.
Are we staying in this room?
Yes, we are staying in this room. So we continue the schedule on 11:15 here in this room. Yeah. Thank you very much. See you soon.
Hello,
Bye.
Mas, we will back at what time, Mas?
Pradina, can you hear us?
Internationale Agrarstudenten. Jährlich ist das auf so einer Konferenz.
Can you hear us?
Yes, we can hear you.
All right, great. I will share the screen
Es sind auch immer trotzdem
so you can confirm if it's working.
Jeder Student hat ja vielleicht vorher auch schon einen anderen Bachelor oder sowas gemacht und das macht halt, finde ich, sehr, sehr spannend.
Can you see the presentation now?
Natürlich, weil wir auch mit der FAU zusammenarbeiten.
Great. Let me check.
Viel gesponsert haben.
You see it going to the next slide now?
No. Yeah.
All right, perfect. Can you spotlight my phone? So then you see the person speaking next to the presentation.
Ja. Und nicht einfach nur das Event stattfindet, jeder nimmt irgendwas mit nach Hause, sondern das ist da so eine allgemeine Message.
Hello, Mas.
You see? Yeah?
I spotlight both of them. Your phone and your laptop.
All right, that's good. That's good.
Is it working?
Yeah. All right, then I think we're up and running, Ferdinand.
Yeah. I'm working on a replication model for.
Right. All right. Good.
Trying to use and our problem is that we're using a satellite, as a reflection of 30 by 30 years. So there's a lot of.
Aku belum buat Milo.
What's up, Ferdinand? Yurika, what's up?
Yes. No. I talk with my brother. Sorry.
Okay. Oh, good. Oh, good. I will put the audio off now, so we can't hear you here anymore. If there's a problem, you have to say it in the ICI in Bonn chat. Yeah.
Okay.
All right.
As far as I know, so they can some work of the most we have a fraction of cutting down the list of 10% but how it's the first area. And
yeah, so that one is checking. I will grab my charger so that the microphone
can stay here.
Yeah, I think we'll hand out some money.
So you can check the work coming to the work in the Cultivar.
I think we can.
We're also trying to couple with airborne diseases one year. So we would kind of take like documented pictures so that we have the pictures of only olive meat and we're trying, yeah. I mean, with the airport, yeah.
Yeah. You don't want to interrupt.
Yeah,
what do you try to separate? What do you try to classify? So what are you doing?
Yeah.
Are you Thomas? Okay, I'm Tom. Yeah. And I was just talking to a few persons who would they need a PC artist for their journey tomorrow. And because they need them tomorrow, I think they have to do them today until 1 p.m. Because the artist station in the market is like it's saying that they need to do their test until 1 p.m. to get the result today. So you want to join them? And I don't want to join them. I have a Mac that they just will be here in maybe 5 minutes to discuss this.
What do you try to? What do you try to?
Well, we're actually trying to find a way to
So we can make an announcement before the presentation.
because you're working with the Japanese, they don't know where all of these are.
And I would have said that they should catch a taxi to be there because maybe it's easier than if they have to take a bus somewhere and the taxi maybe they all share a big taxi then maximum 20 euros.
They get the test but they need it tomorrow.
Yeah, but it's valid for 48 hours, I think. And tomorrow morning somewhere, then the result will be there Sunday evening.
Yeah, so it's not they should close this.
Not closes, but closes for PCR test on the same day.
Yeah, but when they get PCR result, it's Sunday. But if you think about your
Sonntagabend. Also mixing oder ob das geworden ist auch schon, also die Ergebnisse rausgeben. Also ich habe hier Corona-Folgen ist das am Markt. Ich habe irgendwo noch kein Test gemacht. Aber am Markt ist das.
Pixel with the bad resolution, and then you can only use the spectrum.
And you use in the same day PCR test, Bonn, Mark. Ja, das kostet
100 farmers with
yeah, so yeah.
There's some work on the board author. Yeah, there was some work which shows that especially in are beneficial. So it's so on to the other side.
Ja, von der Webseite 1 bis 2. Ja, so they have
pushing him a little bit.
I don't know how much the organization with that because
but they are asking me this would be also, I guess, so somebody mentioned that if we have, we can send this information to the application also with they should respect for the because I will not put exactly yeah, but you can call Germany.
Yeah, I call the test station and tell them if they are
I will leave you for the coffee.
Yeah, at least we know that they are.
So you have some overlap.
Yeah, what did you want?
So they show the. Okay, I will tell them. Yeah, my computer just died. Moving towards you in my backpack without any reason. Oh, it's really warm. Oh, let me open it. Oh, let me plug it into electricity.
Oh, really? That's a
yeah.
problem.
Oh, it's it is. But
don't mess because I think
oh, okay.
made up the internet connection.
Oh, what I showed is this phenotyping concept. But you also have here, okay, these 5 pillars, okay, example of growth, a study of an example of how to use accumulate data that you may get close to a biomass prediction. Example of three-phase, and I showed the video also from the positioning systems. Okay, so I can refer to these things because you already have it. But you also have some more thought. I didn't go into the background of photosynthesis, so that actually connects nicely because there's also the way I have much material. Situation. It's like what you're planning Alle directions. Yeah, I go in other directions, but there but yeah, but that's the fun part. I sent you the stuff already. It doesn't do anything. So, but I only posted this morning. I survived because genau, danach hat der Markt interessiert. Das ist auch dabei, dass die Leute okay, genau. Okay, guys, please listen. We want to continue. Oops. Okay. Hier. Ja, aber das ist okay, now so we continue with our next speaker, Professor Rascher. He is
leader of the Shoot Dynamic Institute of the Forschung Center Jülich, where some of you have been yesterday. And he will talk about the digitalization of trophic systems, the way towards more sustainability, and improved consumption and exploitation. Stay tuned.
Thank you, Jan. And I learned as long as I stay back here, I'm allowed to take off the mask. Good. So, thanks for having me here. Thanks for inviting me. And I'm glad to see that you are also slowly coming out of the Corona lockdown and meet in person again. So it's good to see you in 3D and in reality after 2 years of teaching online only, discussing online. So I'm really glad to be here. And what I would like to do with you in the next hour, and we said we have approximately an hour. I think lunch starts at 12, so we see how much interest is there from your side and how much time we need and how hungry you are at the end. And Jan challenged me to talk about digitalization. And then on the other hand, he said, well, but you're also the future scientist and you can also think and you would like also to dive into science and some scientific topics. So I will try to cover both. I will talk about digitalization a little bit more in a broad sense. And then I will pick out one part of the whole topic of digitalization and tell you a little bit more how the development of sensors, sensor networks, data networks actually happened on this example. To then at the end move to a global scale and talk a little bit about satellite images, data which are already there, how they are distributed, how they potentially can be used now and in the future. So this is the idea for today. Interrupt me if it gets boring. If things are totally unclear, raise your hand. We're in a small group and we can discuss openly. So don't be shy, stop me for any questions. So, as I said, I just dive in. Digitalization, it's the one part I would like to talk with you about. And now let's see. And let's go back. Exactly, and the other thing is then about measuring plant function across scales. And I will tell you soon what's behind there. So let's speak about digitalization or digital revolution. So if you Google agriculture, future smart agriculture or digitalization, I think you will pretty soon come to pictures like this. And I stole a few pictures, which are from Bayer, Syngenta, and some of the global players. And if you look on these pictures, on these visions for the future, you always find the same concepts. You have a farm, which is somehow managed by the farmer. And then there's a full network of sensors, drones, satellite images, ground-based sensors on cows, on tractors. And this all flows somewhere together in a central data network to provide online real-time information from the farmer how to manage the field. So this is kind of the vision which we currently, I think, have, or at least I have in my head, and maybe you also, at least the vision of the high-tech countries. I think as soon as we already move in other countries, the vision for digitalized agriculture of the future will be totally different. But here in our region, this is kind of the way how digitalization is often seen. And what I would like to, something is exactly, and if we think a little bit more, what are the elements of these things? The one part is that in the meantime, we have a lot of novel sensors in hand, which were developed in the past decades, I would say, which give us now real online information on plant structure, plant function in real time. I think Onno already talked about this sensor a little bit, and some of those technologies I will build up on these things, hopefully not repeat this. I have an idea of what he talked about, but just to link up. So we have a lot of sensors, and I also will talk on one specific trait later. But then we also have a lot of new sensor positioning systems. We have drones, little unmanned aircrafts, but also satellite images, which become more and more available. And if you look further in the vision, in the future, we most likely have the smart small machines. Not those big agricultural machines anymore, but the idea is to more and more provide small little robots which find the region of interest by themselves, which navigate independently, which sense the plants, and which also can do some acting on the plants. For example, localized spraying or removal of weeds. This, at the moment, is still under development. I think some are already on the market, but it's more on the tech freaks, it's more on the toy part, and it's not used or implemented widely in agriculture. And then in the background, there's another big technology happening, which is not so visible because it's not machines, but it's the way how to handle data. And you all have heard about this artificial intelligence, machine learning, deep learning, where data from different sources are now combined and used for a guidance, not only for self-driving. That's where a lot of funding is currently going in to provide this autonomous or self-driving, but also for agriculture. And I think you know better than I do all the guidance system and the GPS assistance systems which are already implemented in the bush systems or in modern agricultural machinery. And all these aspects normally come together if we talk about digitalization in agriculture. And I think there's even more. We should not forget everything which is behind or after plant production, in terms of logistics, management of flows of material, and so on. So it's not only about the whole production of agricultural products, but also the usage and the distribution. So that is when we talk about digitalization. And it's, I think, a lot of these things are not only buzzwords, but I would assume are really actually happening in reality. So, and now let's go a little bit more into depth in terms of like, what does it mean to build up this digitalization in the whole big thing? If we think about what does it mean to measure plants? What does it mean to know or to learn something about the performance of plants or the products of plants? And one topic, one buzzword, which you may have heard, is this phenotyping, plant phenotyping. I think that's also in the media a lot. And if I ask you, like, what's plant phenotyping? You know it because it's in one of the lectures, or in the people who learned at Bonn University, I think you know this already. So plant phenotyping is the idea or the concept to measure the traits, the properties of the plants in space and time, and to understand the dynamic development of the properties on the background of the genetic background of a plant. So which variety, which genome does a plant bring with, and to which environmental conditions are the plants exposed. These two factors interact, and these two factors at the end define how the plant product actually is, how big it is, how much yield it has, how much biomass, how other components are in there. But it's not only about yield, it's potentially also how much of a resistance against specific stresses does a plant bring with. Is it resilient against specific pathogens? Yes or no. So this is the plant phenotype. And if you look a little bit back in terms of what you have learned at university and where the research funding was going into, in the past, I would say 10, 20 years, most of the funding went into the genetics. We wanted to know what is really the genetic pool of plants. This is pretty much widely accepted and widely proceeded that we know the genetic material of all our major crops in the meantime, at least of the Western ones or of industrial worlds. And now a lot of development was going actually into this phenotyping, understanding the phenome of the plants. And this is something I brought you some movies, but we decided those movies are not running. Unfortunately, it's a Mac PC problem. We can try again, but it's not really running. But I think those of you who have been in Jülich, they saw these instrumentations yesterday. You walked through the greenhouses, and I think you saw some of those installations. And what we've seen in Jülich, that's some of the developmental platforms, which in the meantime are also implemented at the big players at Bayer, Syngenta, BASF, and so on, who also do this phenotyping, not on a research level, but really on the commercial or plant developmental level. So, not sure somehow, I still have some. Okay, means laboratory setups were yesterday. Let's go to the field. Especially field phenotyping is important because if you want to understand the interlink between genotype and the environment, you already have to go into the field. And if you want to then think about, or if you work in agricultural setups, of course, also management comes in, and you always, for the phenotyping research, we try to understand what is the interlink between the genomic background, the environmental interaction, and the farming management. And you have seen this before. That's what Onno showed you. This is a little bit our mantra, where we say, what do you need for plant phenotyping? And what I would like to go with you is not necessarily in the positioning systems. I think this Onno has covered already quite a lot, but I will come back at the end a little bit to the positioning systems, and everything which I tell you today or in the next half an hour has not necessarily something to do with the positioning system. The positioning system is not a sensor. A positioning, a drone does not measure anything on the plant. It just places the sensor on the right position at the right scale to derive the correct information. So the positioning systems are important for getting the right scale. And I will come back at the very end where I say, sometimes it does not really matter if I measure from a drone, from the ground, from the aircraft, or from the satellite. There are some entities, some measurement approaches, which are pretty much the same and which can be scaled across these scales. I just had a discussion in the coffee break about finding olive trees on a 30 by 30 meter pixel satellite image. I don't see her at the moment, but that's one of the typical examples. I will come back at the end of the talk. So let's talk for the next 20 minutes about another relevant trait, and namely photosynthesis. I think at the discussion with Onno, you covered photosynthesis briefly. Let's think about photosynthesis. And why photosynthesis? So photosynthesis, I think you all know still from your university, this basic biochemical, biophysical process which makes the world spin. So plants absorb light, fix the energy of the light, or stabilize the energy of the light, which enables them to fix carbon dioxide. And this is the basis for all plant material, which is finally produced. The basis for all this primary productivity on Earth is the process of photosynthesis. And when I started to, when I just was a young postdoc, was about 20, 25 years ago in the US, there we talked about the people at the Carnegie Institution, which was an association with the University of California. And I thought, oh, wouldn't it be nice to be able to measure photosynthesis? Some of the people said, oh, this is the mantra of plant biologists and plant production, and the holy grail on the other side. It would be so nice to be able to measure photosynthesis. In the meantime, I think we are one step further to measure photosynthesis. And the problem to measure photosynthesis is that the process of photosynthesis happens at an extremely small scale. So if you zoom in to a leaf, that's where photosynthesis happens. You find the plant cells. In the cells, the little organs, the chloroplasts. These organs have two membranes. The inner membrane is folded and builds these stacks, which are called thylakoids. And in these thylakoids, which are a double membrane structure, in these thylakoids, you have this blob of enzymes, the so-called photosystems. And these enzyme blobs, they absorb light, and they are the first step to actually stabilize this energy and provide the energy for carbon fixation. It's even more complex. In the most textbooks, you do not look in the structure of these photosystems, but if you do so, you find a pretty complex biochemical backbone in which approximately 400 to 600 pigments are held exactly in place. And these pigments, divided like harvesting complexes, core complexes, are built as antenna to harvest the photons and to efficiently stabilize this energy. That's one of the processes plant biologists had fun for the 40, 50 years to unravel and to understand how this actually works. About 12 Nobel Prizes went into the discovery of this whole system of photosynthesis. And in the meantime, we know exactly where every molecule sits. And we know exactly if you have a change in one subunit, what does this affect in terms of photosynthesis. So the process, very small process on the pigment level, is extremely well understood. It's extremely small. So I'm now down to the nanometer scale. And nevertheless, you would like to measure this photosynthesis because it's also on a larger scale, the basis, what we said before, for all biomass production. And also on the other hand, if you come from the other side, if plants are being stressed, if they are in drought stress, if they are affected by pests, these external threats or these internal factors normally leave their signature in the photosynthetic machinery. Very simple. If it's too dry, photosynthesis does not work very well. You know this as a farmer pretty well. Plants stop to grow because the stomachs close and photosynthesis is down. Means pretty much all those extra environmental factors also are seen in photosynthesis. And one holy grail was, can't we measure the dynamics of photosynthesis? And then we can, on the one hand, predict how good plants grow. And on the other hand, we also have a sensor, a universal indicator for plant stress. And this is the science story, like how do we get from this holy grail of measuring photosynthesis to really a system where we can potentially do this from the field up to the satellite in the future. So how do we do this? The way what we do is we again look in the small scale biochemical process of photosynthesis. And if you look in the first steps of how light is used by plants in this blob, in this pigment blob, there, if you look in a textbook, you will see a picture like this. So you have the photosystems, schematically, these pigments, they harvest light. Light is absorbed. This means the energy is used to shift some of the pigments in an excited state. From there, the energy is transferred to a second photosystem where again light is absorbed. Another excitation happens, and at the end you have biochemical energy which enables the plants to fix CO2. And that's enough with biochemistry or photosynthesis. But this is pretty much the nutshell of photosynthesis. And what happens there, if you look in this biochemical photosystems, then you see that by absorbing light, they also emit another photon, the so-called fluorescence photon. And this is visualized here. I don't know if you can see it. This is a solution of chlorophyll. We shine light on this solution from the one side. And if you look carefully from the other side, perpendicular, you see a faint red glow, which always occurs if the plants absorb light in their photosystems. And this is one of the first things. So if somebody asks you, when you go out of the rooms, what color plants have, yes, they are green. This is something to do with absorbing light. But if you are able to look really carefully, then you would also see that all plants glow faintly red. There's always a faint red glow behind those plants, which you normally cannot see with your bare eye, but you can already make this visible by using some colored glasses, by separating the blue light from the red light. The red glow is always in the red, and then this red glow can already be made visible. I don't know if somebody has seen, just before Corona, we had an exhibition in the Deutsche Museum here in Bonn, where we actually demonstrated this red glow of the vegetation, first in the Botanical Garden in Düsseldorf, and then also here in the Deutsche Museum. I don't know if anybody had been in this exhibition. Kind of fun, you could walk around with a blue flashlight, and with the red glasses or yellow glasses, you could see that the plants suddenly turned red. Artificial plants don't do this. They are only green. Artificial grass doesn't do this. And the nice thing behind this is, if you observe this red light, you have pretty much a look into the functioning of the photosynthetic machinery. Because this is a side product. Whenever, let's go back to the scheme which we had before. So whenever, same thing which I showed you before, when plants absorb light in the pigment apparatus, this light normally is used, shuffled to the reaction center, and then the energy is used in photosynthesis. This is the concept which I showed you before. However, even though plants are really efficient, they have an efficiency rate of 83% in maximum, that's pretty cool, but not all the energy of the reaction center can be used in photosynthesis. And energy, which is somewhere in the reaction center but cannot be used in photosynthesis, may be re-emitted as this red glow. And this is fluorescence. Physically fluorescence, light is absorbed and then converted back, and the energy is emitted as a fluorescence. And the nice thing is, this fluorescence signal is variable. It changes depending on how much light goes in, how much energy goes out, and how much non-used energy you have in the reaction center. And this non-used energy determines the intensity of your fluorescence light. So pretty simple. If you have a block in photosynthesis, because the plant is stressed, then the fluorescence signal goes up. In the first instance, it becomes a little bit more complicated, but at the first instance, this is the relationship. And this is something which plant biologists use for decades. Discovered was this process about 120 years ago by first physicists and plant biologists in dark lab rooms. And then in the next years, there were a lot of instruments being developed to measure this fluorescence light as a proxy and an estimate for measuring photosynthesis. The instruments are fast, they are non-invasive. You just use a leaf clip, you measure this red glow, and you get an idea of the intensity of photosynthesis. I think Onno talked about this thing a little bit. Did he show you this data? So back when we tried to find the Holy Grail to measure photosynthesis in the United States, we thought, wow, wouldn't it be possible to get or to transfer these instruments where you have to clip on one leaf and measure the light and the glow directly at the leaf? Wouldn't it be possible to go a little bit away and to measure this red glow, this red fluorescence light also from a distance? With people from oceanography, we designed this monster, big telescope with a big laser. And this laser was able to shoot at this time up to a distance of 20 meters to a plant canopy, excite the fluorescence signal, excite and modulate the photosynthesis, and then to measure from some distance the fluorescence light. You could also modulate the light. This was needed for getting a deeper information of the photosynthetic machinery. Don't worry about this measurement protocol. But in essence, we could measure, and this is a tower at Campus Klein-Altendorf, from 20-30 meter distance, we could also measure this fluorescence light. This was the first development, but still a huge, monstrous apparatus, which most likely at this time was also not fully eye-safe. I think, nevertheless, we operated. Then there was the next step of measurement. After the proof of concept was laid, so it worked in principle, together with some other colleagues, we went into a miniaturization of this measurement instrument, and this huge monster finally was reduced to a size of approximately this projector, a little bit bigger, but in principle, same measurement instrument. You shine light, modulate light on the plant, you activate photosynthesis, and you measure the emitted fluorescence. This was something, I think that's where Onno talked about, where in the meantime, we are at a point that this instrument is so mobile that we can transport it across the world. You don't need a complex measurement or positioning system to measure there. And I think Onno also talked about Nicholas, who ran in the USDA over the fields to measure really plant photosynthesis from this big 3D plan. To give you an idea, until we arrived there, I would say this was approximately the intensity of 10 PhD theses, instrument development, measurement protocol development, understanding of the signal, and so on. 10 years, 10 PhD theses went into until we had this little instrument which can be used operational still in a research phase. So, having said this, we nevertheless were not really happy, or we're not satisfied with this, because we would like to measure plant photosynthesis even on a larger scale. There were attempts to put this laser on an aircraft. People also thought about to put this laser on a satellite to shoot from space this active pulse on Earth, but this never materialized. And if you think a little bit about it, you also will realize maybe not a good idea to shoot a really modulated laser on space on plants to excite photosynthesis. But nevertheless, we wanted to go to the next scale, namely the passive and global measurement of photosynthesis. To do this, or to achieve this, we tried to use a measurement trick. I think Onno did not talk about this at all, isn't it? So now, next trick, and this went back to a Russian scientist who also knew this fluorescent signal, and he thought about a measurement principle and concept in the 70s, and also published this in Russia. Nobody actually at this time recognized this. But the idea was that if you measure from out of space, you have, if you look on the light which comes on the, which reaches the earth of the, which reaches the surface of our earth, then if you look on the wavelength, means the spectrum of the incoming photons. So this is the wavelength, that's red light, and this becomes far red, you barely can see this. And if you look on the transmissivity of our Earth atmosphere, which is the blue line, then you realize that there are some small so-called spectral windows where the incoming photons become absorbed by the Earth atmosphere. And this, for example, these two bits, which you see here, is the oxygen of our Earth atmosphere, which interact with the incoming photons. And incoming photons are actually absorbed in the Earth atmosphere if they have exactly a wavelength of 760 nanometers. They penetrate through the Earth's atmosphere if they have a wavelength of 750 nanometers. So it's a very wavelength-specific absorption in the Earth atmosphere. And the measurement principle now is, if you consider that the fluorescence light, which is here seen in the green light, is still emitted. We talked about this before. It's a glow, so the plants glow, so it's still emitted. And then the concept was, if we are now able to measure inside of these small absorption lights, we have no incoming photons which are reflected or which go to my sensor, but the plants are nevertheless glowing. And if we are able to retrieve and to resolve these small absorption lines, then I should be able to see this red glow of chlorophyll. And this was the measurement principle, the so-called Fraunhofer line principle. And in essence, that's a little bit more schematically. You have these absorption features. You know that you have an added fluorescence signal to the back. And to resolve this, you compare your intensity inside of the absorption features and outside. You pretty much measure the depth of this band and the shift of this band depth. And by doing so, you can separate reflectance and fluorescence. That's schematically the background. If you're interested in the mathematics, you can also look up the mathematics. It's kind of handleable. Technically, you must be able to resolve this small band absorption lines. And this was then something where people first started to do this with high-resolution spectrometers, which can be built on the ground. And that's another kind of side story why we have developed this lift laser. Other people said, okay, can we build high-resolution spectrometers in a rigid box, put it on a tower, and permanently measure passively the so-called solar-induced fluorescence? Again, science story. Maybe not 10 PhD theses, but 7 or 8 PhD theses went in. It was a co-development with the University of Milano and our side. The first instruments looked terrible, wooden boxes with cables hanging out. And then at the end, we managed to put this approximately in a measurement, waterproof, environmental proof box like this, the speaker. This then was a company which went out and is now still operating on the market. So JP Hyperspectral, the two owners were a little startup in Neuss in Düsseldorf, PhD student from my group and a PhD student from Milano. They both own this company and they built the systems in the meantime. It works operational. Everything is built in the boxes in the computers. And I think we have at the moment 45 of those boxes being operated worldwide, distributed on towers like what you see there. That's Spain, that's a field site on our side, but they are also now distributed worldwide. And we recently have taken the first box in operation in Africa, in Central Africa, to measure continuously the fluorescence signal of vegetation. So momentary measurement to record and to measure fluorescence as a proxy of photosynthesis globally. Well, on some points which are distributed globally. We are not on the global level, but we have a measurement box which can measure this permanently. I think I skipped, let me think, yeah, I think I skipped these things a little bit. And let's go to the next part. What I wanted to tell you there is just for you, if you were really interested in this fluorescence signal. I was lying to you a little bit because the concept which I showed you before with light coming in, photosynthesis using the energy, and the fluorescence being a proxy of the energy which is used, which is not used. This was true, but the problem is if photosynthesis is blocked, if a plant, for example, has stress, then the plant is pretty clever and avoids that there's a huge amount of energy being accumulated in the leaves, in the photosystems. So the plant normally activates a whole set of non-photochemical dissipation processes or protection processes to get rid of this excessive energy. And this third process also sucks out energy of this reaction center. And also, if this happens, the fluorescence signal again decreases. Means measuring fluorescence is nice, but it's not a simple one-to-one relationship with photosynthesis because we have a three-part process, and means for the understanding of the fluorescence signal, we always have to keep in mind that we have different phases. So this is a relation between, this is the efficiency of photosynthesis, this is the efficiency of fluorescence. And what you can see is that it's not a simple one straight line, but that you have different phases where you have one phase, which is normally the plant is pretty happy phase. You have this linear or antilinear relation between fluorescence and photosynthesis, no NPQ. You have one part where this additional third mechanism kicks in, reduces the fluorescence again. And then you have a third part, which is an extreme stress when all these dynamic regulation processes in photosynthesis do not work anymore. Then you have really an accumulation of electrons in the reaction center, and then fluorescence increases again. But on the other hand, photosynthesis decreases because this is the pathway of dying, severe stress, severe limitation. So just for keeping in mind, fluorescence and photosynthesis is not a simple one-to-one relationship, but we understood the mechanisms behind there because we have all this understanding of what happens in the photosystem. So, and this also works on the larger scale. And with this, back to what I wanted to say, let's go back to the boxes and to the scale to measure the solar-induced fluorescence also globally. And the next step was to see if this is possible to measure from an aircraft. So we're moving it further a little bit away, up to an aircraft. And the idea was, again, to build a sensor which is able to resolve these absorption features, which I showed you before schematically. Because if you're able to do this, that's again the wavelength. If you're able to resolve these absorption features, there we can see this fluorescence. This was a challenge which I went out to industry about 10, 12 years ago. And I wanted to have a company somewhere worldwide to build such a sensor which can resolve this absorption features and which can be mounted in an aircraft. And then it got a lot of frustrating responses. Most of the companies told me that's totally impossible, technically not feasible. Other companies wanted so much money which I could not afford. And then finally, I found one company somewhere far north in Finland, Specken, which has extremely good optical engineers. And they said, well, we have something in our drawer and we tried this, we can try. And then it was a 5-year of development time. The company hated me, I think, at the end, because they threw away a few sensors on the way. So they built a full-fledged sensor, we tested it, we realized, no, not good enough. And then they started over and over again. But at the end, by 2012, we got this sensor delivered, which we called HiPlant. And this sensor, this is now data really from flying, was able to measure this small scale features from a moving aircraft. Another story, sensor development. This sensor in the meantime is operationally available. So the company, I think, lost a lot of money with my contract because they had to develop it first. But in the meantime, 7 of these sensors are flying worldwide. So they are also now sold to different countries. Interestingly, 2 to China, where I only know from 1, which is flying, and assume the second sensor is being ripped apart, and that some Chinese companies try to understand what did they do technically in this sensor. But nevertheless, they are now flying also on all different countries. And what can we do with this sensor? So just a few snapshots. Use the same retrieval as before. So we look in the absorption lines. There's a lot of further development behind to move and to correct for this wobbly aircraft and to, at the end, get airborne data. But what we at the end can receive are images like this. This is the Forschungszentrum in Jülich. We visited our institute that's up there. And this is a pretty mixed agricultural area. When we drove up, you saw this, I think, yesterday, a lot of intensive agriculture on good soils. And this is from somewhere in the summer, where you can see the small scale patch of different crops, which not only are mapped according to the greenness, but they are now also mapped according to the intensity of the fluorescence emission. So this is greenness plus, and additionally, activity of photosynthesis. And the resolution of the sensor, or that's how we designed it, is that we can also, for the other group who was at Campus Klein-Altendorf yesterday, if you look at the Campus Klein-Altendorf, that's a mapping of the Campus Klein-Altendorf, you see the different fields at Campus Klein-Altendorf, and we can subsect those fields in the meantime according to the different crops. And this is a classical field design with 3-meter experimental plots, and that's what we can resolve on the lower basis with our sensor. So it's a pixel resolution of 1 meter approximately from the aircraft. We know where we look at, so we can pretty much go from the experimental plot design, 3 by 3 meters is our limit, up to the whole region of several kilometers. Few examples. What can we do with this? Another picture close up from Campus Klein-Altendorf. But what we have done at the very beginning, this was, I think, 2013-14, one of the first experiments where people were not really sure of what does this fluorescent signal really mean? What do we measure with this? We made a pretty simple experiment in the field in the Czech Republic, close to the Slovenian border, where we used artificial natural grass, but Rollrasen. So you roll out this normal grass lawn. You then have 2 areas, and one area we treated with DCMU. Who knows what DCMU is? Diuron herbicide. Still used in South America, blocked in Europe because of the interference with bees, but still South America uses it quite a lot, and it's a photosynthesis blocker. So it kills the weed by blocking the photosynthetic protection apparatus, the yellow arrow which I showed you before. The plant cannot get rid of excessive energy and dies of sunburn. That's the mean of operation of this diuron. And what happens, and this we know from the laboratory, is if you apply diuron, so you block photosynthesis, and as a reaction, the fluorescence signal greatly increases because you block this normal natural pathway. And that's what we did in the field. So one side, we sprayed diuron, and then 1 hour later, we flew over this area. And what we saw is that in the normal NDVI greenness index, so there we only look on the greenness, we didn't see any change. But 1 hour later, we had this great increase of the fluorescent signal in the sprayed plants. So no breakdown of chlorophyll, but a block of photosynthesis. It's dependent on the dose. So we have different doses. It's a little bit dependent on the timing, but we could show that this is really a functional information where we could look into the functioning of the photosynthetic machinery. And this then was used a lot in agricultural parts to, for example, use some agricultural fields and to relate this signal to carbon uptake, GPP, gross primary productivity. So this is now on the other part, an estimate of the GPP, which you see here, CO2 uptake, and we tested if including this fluorescent signal actually improves our prediction of carbon uptake. Well, that's what most likely important. As a farmer, you're not interested in fluorescence, but we use fluorescence to estimate, for example, carbon uptake. And we could show that this model could be improved by including this fluorescent signal. We made some more measurements. That's an example from Italy, where we tried different herbicides and different treatments on the ground and tried to understand what does it actually have as an influence on the fluorescent signal and the growth rate on the ground. We played with weird plants. That's a genetically modified soybean. which was developed by the University of Illinois, which has a greatly reduced leaf chlorophyll content. So it looks kind of pale, but it grows fine. It produces 98% of the biomass, besides having almost only a third of the leaf chlorophyll content. And we were all, for quite some while, puzzled, how does this plant actually do this? Because normally we would say chlorophyll is important for plant growth, and this plant does well without chlorophyll. And we found some understanding of what's behind there. And we also went into some studies where we covered the European heat waves in the past years. So this was the heat wave in 2015, but another where we try to see if we can see the impact, the immediate impact of a heat wave on the momentary growth. This was always the idea, to not only wait till the end of the vegetation period where you see, well, we have a reduced yield. Well, most likely it was because of a heat wave, but to see already the downregulation of photosynthesis while extreme events happen. And this was also something which, this is Campus Klein-Altendorf and people from Austria could also. I skip a few of these things because I think we also need a few more time for discussing. You get the slides later. I put you in the paper. So if you're interested, you can read the papers or contact me. But what you may have read or seen there already is that all the work which we are doing now, which I mentioned, was not only be done here anymore, but this was all linked to some international collaborations. Some we did in Italy, in Switzerland, Czech Republic. And this was fueled by then the interest of the European Space Agency to test if a satellite could be built, which measures the same thing like our little ground box or our aircraft. Sensor technology was better 7-8 years later, and with the millions of the European Space Agency, we went forward and developed a concept for the so-called FLEX satellite. FLEX, the abbreviation for Fluorescence Explorer. And the concept was, well, can't we measure now really globally the fluorescent signal from space from such a dedicated satellite? That's a picture of Campus Klein-Altendorf again, down here. And this is the official logo from the European Space Agency, which made you think already that at the end, after several rounds of evaluation, we were successful. And now the European Space Agency is building a satellite in the branch of the Earth Explorer satellites, which will be launched in 2024. And I'm now on the satellite scale. So let's talk a little bit about satellites. If you think about European satellites, you have a big branch of meteorological satellites. You see that at the moment, there are approximately 15 satellites operated by ESA, European Space Agency. Almost half is for observing weather. Then we have an operational scheme or an operational branch, which are the Sentinel satellites. I come back to them soon, and you may have heard this. Sentinel-2 is the major agricultural satellite, but they are numbered. At the moment, we have Sentinel-5P in orbit. So Sentinel-1 to Sentinel-5 are operational. And then there's another branch of satellites, which are the scientific satellites, which are not geared towards the end user. So it's not that you have free data there at the end, but here we test new measurement concepts, which then in the future, when I'm most likely retired, potentially, if successful and if useful, go into this operational observation of our Earth. And there we are there with this fluorescence sensor at number 8, being launched 2024. And if you sometime in 30 years or so download fluorescence data, then maybe you remember the day today. That's maybe the first time when you already understood what's good. How does such a satellite mission work? Our FLEX sensor is a little bit novel. Additionally, as we are trying to implement a new measurement concept, where we follow at a time delay from 6 to 15 seconds another satellite, Sentinel-3, that's one of the operational satellites, and we try to follow exactly Sentinel-3, look at the same surface area, and the advantage by this is that we can link the data, so it's a big save of money that we can use some of the data which are provided by Sentinel-3, which we also need for retrieving the fluorescence signal, because we have to solve all these atmospheric issues in between. So the data flow of Sentinel-3 and the data flow from our experimental satellite are linked, and both used then on the ground for data processing. What we will go with Flex is that it will be a global mission. It was a long discussion where should the satellite look at, and at the end the community decided that it should be global to really cover the whole Earth. It's a maximum of every 27 days, or in Europe it will be every 10 to 15 days because the orbits are overlapping, and it will have a resolution of 300 by 300 meter pixels. And there you already realize something. This is also a compromise about how much money you have, how big can a satellite be. The best what we could measure with the scientific mission is a 300 by 300 meter pixel. This means, be aware, we most likely don't see single fields anymore by this. Or if you are in South America, then this could be possible. What happened then? In parallel, while ESA decided to build a satellite, other peoples worldwide made some thinking and thought about, well, can't we use the existing satellites, which are already in space, and retrieve this fluorescent signal already from the existing satellites? And this is the first picture which people from NASA, Joanna Joyner, published, and she used an atmospheric satellite. And she was principally interested in the composition of the Earth atmosphere with the focus of oxygen. If you can remember, these absorption lines, they have something to do with oxygen. And then Joanna contacted us and said, well, I always have this error and this noise in my data when we fly over dense vegetation. And then I said, well, maybe this is that you see this fluorescent signal from vegetation. And then we had some discussions. Joanna also tried to understand what fluorescence is. And she finally reprogrammed the satellite and could use this original atmospheric satellite, GOSAT, to measure the atmosphere, to use the data and to look at the noise, which form was noise in the atmospheric sciences. And she could then, for the first time, also retrieve fluorescence from this existing satellite. And this then finally triggered that several scientists worldwide looked on the existing satellite data. NASA actually redesigned one satellite, OCO2, slightly and redesigned the optical properties to also be able to measure fluorescence from this existing satellites. And if you look on the web, if you have fun after this, there are a lot of movies and data and pictures also now from NASA, where you can see in an animated way our Earth in this representation of the fluorescent signal means the indication of where and when photosynthesis is active. I just downloaded a few recent data. This is the Nile, with the big Nile data, big agricultural area. And from the existing satellites, you can already see the different areas of vegetation of when you have active farming in this area. Currently, we are at a scale of about 3 to 5 kilometers of a pixel scale, and we will then further improve this, as I said, this Flex in two years from now. So, and with this, I'm almost, no, I think not almost at the end, I am at the end. There are a lot of web pages, of course. Behind a satellite mission, there is always a lot of material also be found on the web. I think you should know all the buzzwords now behind that you can have fun to look at more of this data. What I would like to encourage you also is exploit the data of the Sentinel satellites, which are now available by the European Space Agency. The European Space Agency has built, I think, with the Sentinel family, the best satellites ever humankind has had. So from the optical performance, it's an extremely good new fleet. And the European Space Agency has made the big step that all these data are now freely available for everybody. This was different before, but now you can really just download these data. And I think we are somewhere down here. Bonn, Venusberg is down here. And you can, in the meantime, with easy portals, and that's just a snapshot of the download portal with a recent image, just download different raw data or also reasonably good products like water indices, soil indices, and so on. There are also, wherever you are, there are normally also national institutions. So in Germany, there's, for example, the JKI, which provides a specific agricultural portal where they further process the data. So they merge data from different satellites and produce, for example, recommendations, which are copied out, recommendations where you can grow soy beans and so on. So a lot of agricultural material we now make available. And this is also available from other countries. So if people from international look there, South America has a lot of information being also made available for their agriculture. And with this, I already see Jan, we should, I think, come to an end slowly. But I think, hopefully, I have showed you also, it's the matter of scale. You always have to keep in mind. We had discussed this before, but if you are kind of clever, if you use the right signals which are accessible on the different scales, then you can link these data. And for me, in the head, it's normally the continuum of we want to know something about the plants on the ground. Sometimes we have to rely on ground-based sensors, UAV, aircraft, up to the satellite sensors. This also brings me a little bit back to digitalization. That's what I think where a lot of things will happen in the next years. Sensors is one part. It's fun to develop those, which I most likely have told you that I had fun in my life to develop sensors up to the satellite. But the most breakthrough things will come, I think, in the next years by combining, merging these data across the different scales and over time. And with this, I think you don't have to go to the summary slide. I think I'm kind of at the end. I want to thank all the people who are behind there, because as we have seen, this developing is a lot of effort, not only in my group, interdisciplinary engineers, biologists, agronomists working together, but also across different institutions, which, by the way, is a lot of fun. And thanks again for having me here. And I think I just stop here, and we can, as long as you want to talk about this, have questions or continue during lunch.
Yes, thank you very much, Rascher, for this impactful presentation in the world of fluorescence and digitalization in agriculture. I think most of us, all of us, were really, impressed by this topic. So then we start the discussion. Question, if there are any questions, please. Yeah, there. Can you speak up loud, or do you?
Yeah. Are you familiar with UV equipment, which is developed for killing powdery mildew spores? It's a pathogen. So I would like to know, did it change some physical signals in the plant itself, not only killing powdery mildew, but how it affected the plant?
Yeah. I'm not a pathologist, but powdery mildew I know. It's good. So what I would recommend, there is some work from Ann-Kathrin Marlein. She was formerly at Bonn University, and now she's the director of the Institute of Sugar Beet Research in Göttingen. Marlein is her name, Mahllein, Ann-Kathrin Marlein. And we had two PhD studies together where we tried to first understand what is the impact of the powdery mildew infection on the reflectance of a plant. This is the first thing what you have to understand, because if you spoke about powdery mildew, you have the outer layer which you see, but then you also have the hyphae which go into the tissue of the plant. And both effects alter the reflectance, so alter the way how photons interact with the leaves, how they are absorbed, and which photons, which wavelengths are absorbed. So there are a few papers around this to understand this. When do you see changes in your light depending on the powdery mildew infection? And this is what Ann-Kathrin is also still doing. So she has drones and also dedicated cameras which are optimized for mildew infection. And they are flying this at the moment in Göttingen, at the University of Göttingen. And detection seems to be possible. It's pretty specific on powdery mildew. Then the next part of your question was, can you do some treatment? Did I understand you correctly? That you would also, because that's what you normally do. We try to separate. First, you have to detect it, and then you think about how do you treat it? How do you manipulate it? And in Europe, I would say principal treatment of plants is normally done on ground-based instruments, just simply because if you do this from drones, if you have actors, if you spray, then you also have to carry liters of material, then your drone becomes really heavy and kind of a pretty hazard to the operator. So you're speaking about big, heavy, dangerous drones. And therefore, at the moment, all the treatment in Germany is done on the ground with ground-based systems. And some of the robots which I showed before are currently being developed for treatment. Means the concept would be the drone detects where it is, records the GPS coordinate, and sends a little robot for the spraying. This is at the moment the concept which we mainly do in Europe and the US. The Chinese are acting differently. They don't care about security measures too much. And I have seen dangerous, frightening drones flying in China and doing treatments and spraying. But this was really something experimentally I saw 5 years ago. I don't know if this ever comes greatly to the market, but I would say treatment, I would always do on the ground at the moment. Does this answer your question?
They're not, when I come to it, an artificial intelligence, RGB imaging that could identify voltage view. Yeah, but it's not able to scale it on the satellite or on the drone. RGB imaging.
Yeah. Yes. So that's so there are at the moment two different developments going on. So classically, we use the reflectance data, that is the RGB or multispectral data, and we try to really mechanistically understand what colors change specifically to infection, for example. This is currently challenged by all the people who do this with the machine learning, artificial intelligence, where you just use those approaches from machine learning, you train your network, and then you can also detect how it will do. At the moment, I would say both approaches have pros and cons. There's no clear winner yet, but both are working nicely together. And if you think about the Phenotyping consortium, which we have here in Bonn, we have pretty much both people of both disciplines working together. Second question is about scale. If you talk about disease detection, it would be great to detect a disease also on a large scale from satellite images. And there it becomes tricky, because if you think about your normal agricultural practices, to detect the disease on the ground, you normally look on pretty small signs of the disease. You look on the brown tip of a leaf. You look on the ringling of the main shoot by anything. So that disease signs, and especially the early disease signs, are normally pretty small scale processes, which you can easily detect if you are with your super brain as a farmer on the field. If you, however, scale up, if you cannot see in your image those small properties anymore, if you only have a mixed pixel of a meter or a kilometer scale, normally it's really extremely complicated up to impossible to see the early infection signs, which you normally need to be specific for detecting plant diseases. And this is the problem which at the moment for all the remote sensing parts, I see on the one hand from the farmer side, the need to have satellite-based or large-scale detection of diseases. And from the remote sensing community, they struggle with this. And this is really a limitation of the scale, that the small-scale things are masked in the whole background. Yeah. Was this a little bit?
Yes, of course. This is what I wanted. That's the question.
You said that you use this fluorescence to measure photosynthesis. That you have these three categories, like the initial stress, fluorescence goes up, and then it goes down again. Yeah. But you have similar fluorescence levels for non-stress plants and for stress plants. So how do you differentiate?
Yes. So exactly the scheme which I showed. This is the ideal case if I can control light intensity and the different pathways in the laboratory or in the experimental setup. This, of course, becomes much more complicated if I want to transfer this into agriculture, because there I only see an intensity of a signal, and I do not have this classical control stressed treatment, what I like to have in science. So what there at the moment is in development is that the fluorescence data, which we retrieve from the satellite, they are being provided to the users as so-called level 2 data. This means the Space Agency takes care of all the atmosphere and the georectification and the calibration of the satellite. So you have fluorescence data. But this is not data which you can use as a farmer yet. And that's what I said, it's a scientific mission. So we only, or the European Space Agency will only provide this fluorescence data in terms of intensities. And then the next level, namely that you, for example, look on time series, where you can compare your fluorescence value from this time to the mean which you have observed in the years before, where you can look on the changes in time, where you can look on the contrast with neighboring fields, where potentially also irrigation data or rain data are being fed in, where you use the background information of soil water holding capacity to translate rain data to soil water availability and then make a drought predictor, and so on. So I could continue on this, that you have, for example, on your question, a drought index. You say this vegetation is really drought stressed. We need more than just one snapshot of a fluorescence image. And this is then the higher level data, normally level 3, level 4 data, which are then being provided, for example, by the JKI, by the service providers. So behind their data product is always more than just one snapshot. So on your question, good question. Fluorescence is not the holy grail, what I said before. By measuring fluorescence, you do not see everything, but you contribute with this view into the functioning of the photosynthesis. Second, there are also this two-phase things that comes in. That's then in the models.
Yeah, further questions? No questions out from the local committee. Okay, then I guess your
maybe if you could comment on what you think some other future applications will be from all this data that we get in satellites. What you expect people to do?
So at the moment, you're not talking only about fluorescence, but on the general fleet of all the satellites which we have. Yeah. So what I see at the moment is that the communities which were in the past a little bit dislinked. There was an atmospheric community, they cared about the constitution of our atmosphere and the weather. There was a soil community, there was an ocean community, and there was a land community. And they had their own satellites and their own data products. And these data streams are in the moment flowing together and are used across the different communities. And this gives, for example, a new insight into, I make an example, do you know the drought monitor, which is provided by the Umweltforschungszentrum in Leipzig? It's an online tool, and they produce, for Germany, they produce drought maps, and they monitor the water availability in the soil across Germany. And they do this by satellite data, by soil maps, by modeling, by weather forecast. This product was now extended by a water monitor, where they work together with people from river discharge and vegetation, because it's not only that you look on the soil, but that also you have to think about how much do the plants transpire. And this then further the next step, that there's now a transpiration or water use efficiency product being developed. And I think this is something where a lot of new developments will come. So I think humankind will continue to build new satellites and to upgrade the satellite, the measurement concepts. But we are already, well, it's a little brave, maybe we're not at the end. There will be some development, but we are at some points we are hitting the physical limits already, what's possible. But where we will really see a lot is by using the data across the disciplines using them in time, and also making this data products then mixed, remodeled, available for specific user communities. And that's, I think, what will happen in the next 20 years. And what comes out of there, I don't know, but I'm really surprised by what happens there every 2 years when there's a big conference. So if you have fun with this, you can still sign up, I think, at the Living Planet Symposium, which happens in Bonn as well. So it's in the UN building. It's a free conference, doesn't cost anything. LPS, Living Planet Symposium. And there ESA shows every 3 years what's currently possible with Earth exploration. And there you get an idea of what's available and what is the vision behind the people who build satellites. And as I said, it's definitely next door and one of the things I would highly recommend if you are interested in this.
Very cool, thank you. Okay. Yeah, so now we are ready for lunch.
Lunchtime?
Yeah, we will continue at 2 p.m. So make sure that you are satisfied and you have enough to eat. Just one comment. The dishes for the coffee break, please put it in the gray box outside so that the stuff here is better to handle. Thank you very much.
So yeah, for participants, we're gonna have the lunch break for 2 hours. So we will back at 2 p.m. GMT1 or 8 p.m. GMT7. And we will be having one last lecture. And then after that, we proceed to open mic, which is you guys are given the chance to take over the stage to share with fellow participants your own work, idea, presentation, and stuff regarding the related topic of this event. And then, yeah, you could use the long break to prepare on that. Thank you and enjoy your break.
Pradina, everything all right? Everything all right, Pradina?
Yeah, everything all right.
Okay. We are taking a break now. We'll continue in a bit.
Yeah, taking it for around 2 hours, right?
Yeah.
Sure. We'll be back
Okay.
at 2 p.m. Is it right?
Yeah.
Of
Yeah.
course. Okay.
All right. Mantap. Oke. Sampai jumpa lagi dan see you later. Bye bye.
Bye. Enjoy your break.
Ya, sama-sama.